He's also a visiting faculty member at Harbin Medical University and Wenzhou Medical College. Professor Amir Shahzad has advised and participated in establishing translational medicine infrastructure for several organizations. So please welcome Professor Amir Shahzad.
Hi everyone. First of all, I'm pleased to be here to participate at your annual Congress, and it's a very nice organization. And as it's the first time to be here and to see the motivated students from over the 50 countries, it's a very fascinating kind of achievement, especially by the organizer of the Congress. So I would like to say the congratulations. And along with that, I was going through the scientific agenda, and I realized that even at the students' stage or phase of their studies, so there is an increased awareness to keep updated about the current kind of trends, and to see how the industry and academia is going forward. It's kind of very important, especially for the life sciences the field, the graduates, to keep aware of the everyday kind of changes in the scenario of the prospective, especially if you are going to in industry or if you are planning to go in academia. So there are every day, especially in these days, so there is increased kind of transformations, and the requirements are going to be changed. So I will speak about the basic concepts of the translational medicine. I'm sure that most of you would have some kind of idea about it, but still I'm sure that most of you are not so much familiar about the deep understanding of translational medicine. So I will try my best to explain about the various aspects of the translational medicine. So I'm going to start with the needs of translational medicine, followed by the concepts and the various kind of the definitions, along with the goals and a brief overview of the key strategies and approaches. Afterwards, I will provide you a few examples and a brief overview of the global initiatives and program of translational medicines. So, if we say that why there is a need of translational medicine, so if we say from the scientist point of view, that are working in the labs. So the most of the time, the basic scientists, they are working in the bench, and they are not so much in touch about the industry requirements, and they have a limited kind of interaction to the clinical professionals. So most of the time, so they have performed a high-profile kind of research, and they have come to a point where they have an excellent kind of the data. But as they have the limited interactions, along with other like kind of the reason. So most of the time there the discoveries, they go unnoticed. And another aspect of that kind of situation is that they are unable to understand the clinical requirement and the current industry trends. So it creates a kind of gap in the bench side professionals. So it demands a field that should occupy or cover or bridge that gap. So here comes the translational medicine. And if we speak about the clinical, the professionals, so they are working in the hospitals, and the most of the time they don't have so much time to do the research, and they are not actually good familiarized with the infrastructure of the research and to experimental kind of setup and the techniques. And also along with that, they also have the limited interaction along with the bench, the scientist, and also with the regulatory affairs and industry professional. So it means at the end, the same kind of the problem arises, that there is still gap. So it's not the perfect that a clinician is sitting in his hospital room and he has no kind of the knowledge, or there is a lack of communication with industry, with academia, and with the bench side, the professionals. So here again comes the translational medicine to bridge that gap. And the most important is if we speak about the industry point of view, as you are already familiar that in the last few decades, so there is a declining kind of trend of new drug approvals by FDA or other regulatory agencies. And it has caused a significant the reduction of the jobs and like the budget and the cut of the various infrastructure in pharma industry. So it also creates a kind of gap, and it says that there should be something to overcome that kind of situation. Along with that, if we go to the biotech industry, so nowadays there is an increasing kind of trend for personalized instruments and the diagnostic assays, especially if we speak about the funding organizations, for example, the NIH and the European Union projects, for example, Horizon 2020. So they have a special allocation of the budgets for the research projects that are tailored to personalized healthcare instruments and assays. So to overcome all these kind of the challenges, TM is going to play a very important role. So afterwards, I would like to say that still about translational medicine, there's no kind of definition which is accepted universally. It causes so much confusion, and the various institutions and the various research groups, they have tailored their own concept and the definition of translational medicine to meet their needs. Also, along with translational medicine, so there are the various kind of terminologies are being used. For example, translational biomedicine, translational research, experimental medicine. So all these kind of terminology, they indicate the same phenomena. So they are not like the kind of different things. So if I quote a few examples of the concepts and the definitions, so according to one concept, they are going to refer translational medicine that it's to the clinical investigations with the human subjects, in which the knowledge obtained from the basic the research with genes, cells, or animal models is translated into diagnostic or the kind of the therapeutic intervention with the ultimate aim to prevent or to treat the clinical condition. And according to another concept, so they say that it's translational medicine is an interface between the basic research and its application in the setting of the clinics, especially for the diagnosis, for the prevention and the treatment of the clinical conditions. So there are kind of various concepts. So some concepts are the focusing more on the clinical side, some more on side of the bench. So if I speak about the European EU STM, which I represent here, so we are going to create a consensus on the definition of the translational medicine. So according to EU STM, translational medicine is an interdisciplinary field which brings closure and combines the bench side, bedside, and community to enhance the efficacy of prevention, diagnosis, and the treatment of the clinical condition, and with ultimate aim to improve the global health care. So that the definition is going to be adopted in our like official publications and so on. And we are going to organize a consensus about this, the concept and the definition of the translational medicine. So you should quote that, I mean the definition if you want. So according to EUSTM, translational medicine is a triangle between the bench, bed, and the community. The old concept is that it's a two-way like a pathway between the bench and the bedside. But according to USTM, we say no, it's a triangle. It's going to focus also on the community medicine. The reason why the community, the medicine is important because as you are making new diagnostic assays or tests, or you are making new drugs, but if they are not easily available to the end product, end user, and to the community or the patient, so there is no use, and it's not ultimate improvement in the global health care. So, so we are consider the community medicine as an equal kind of the pillar of this triangle, along with the bench and the bedside. So afterwards, TM, as I already told you the needs. So if we say in few words the goals of translational medicine, the most important, the goal is to accelerate the R&D process and to increase the clinical trial phases and reduce the heavy budgets associated with all phases of the clinical trials. So right now it takes about 14 years from the concept and to a medicine to come in the market. It's a long the process, and it costs a huge amount of money to pharma industry. So it's the main goal of translational medicine is to accelerate that process and to reduce the cost associated in that process. And of course, to achieve that aim, so we need to better understand the pathophysiology of the clinical conditions, especially those conditions that are going to affect the large number of the populations so both in the low and middle income countries and also in the Western countries. For example, Alzheimer, diabetes, and so on. So the main emphasis in translational medicine is to go deep at the molecular kind of level to better understand the pathophysiology. And along with that, as I spoke before, the community medicine is very important. So TM is going to put equal emphasis on the preventive medicine strategies. And already I explained the reason why. And the most important at the end, to make sure that all the applications and the benefits in the form of either diagnostic test, assays, or a new medicine, they arise from the clinics and the bench. They are ultimately reaching to the community. So these are the brief kind of the key goals of the translational medicine. So I will speak about the few the words about the key strategies and approaches that are being used in translational medicine models. So translational medicine is carried out by the combined efforts from the various disciplines and expertise. So if you want to note, so you can note these kind of the tables. I'm going to speak the few words about each point. But if you want to, write this, so it would be very helpful. So I'm going to start with the omics. So omics sciences, in the recent years, it has gained a significant attention by the scientific community. And after this whole genome sequencing, so we have the plenty of information and the data ready to be evaluated and to get the new insight about the various aspects of the clinical conditions and to check their associations at the genetic kind of level. If we speak about potentially the discovery of the drugs, so if we use the omics sciences approaches, for example, the gene association studies, so they are going to provide us a platform for potential new kind of drug, kind of the targets. And also, if we go in deep along with the proteomics and the genomics and the metabolomics, so these are the subfields in the omics sciences. So they are going to provide us the various kind of the potential, the target where we can hit and to evaluate the potential side effects or the toxicity analysis of each compound, and to make a kind of initial the risk assessment of the molecules, and at the same time to explore the various new pathways and to achieve some kind of the consistency to reduce the failure of the molecule at the late stages in the clinical trials. It's the main kind of, how to say, in industry is the main kind of the reason of the failure, and a lot of the budget is wasted when at the late stage of the clinical trial a molecule seems to be toxic, and so it's the withdrawn. So by these approaches, so it's now possible to reduce this kind of a long duration and to save the cost. According to some concept, so they say the translational medicine is just a biomarkers related field. So it's not true. though the biomarkers are the one of the main tools or expertise is being used in translational medicine models. The reason why they are so important that they are providing the benefits in the starting from the molecule from the discovery phase, and they are still continuing to contribute in the clinical phases and also the post-launch phase. So in all phases of the drug, the development process, so they are providing a good insight. For example, as I told before, at a very early stages in the discovery phase, so there are biomarkers that are going to provide the valuable information about the early risk assessments. So these are going to save a lot of the budget and make an early estimation about the candidates. And of course, they are going to reduce the time in the clinical, the trial phases. So there is a kind of trend, especially in the neurosciences field, to use like kind of non-invasive biomarkers. For example, the PET or the scans, MRI, for example. So they are using these imaging kind of techniques to act as a biomarker. And they are proving excellent alternative to the conventional biomarkers. For example, to study the target activity in the presence and absence of the compound, especially in the neurosciences, the drug, the R&D process. And also, a class of the biomarkers is now is being studied that are achievable, or it can be detected in the CSF or the serum, and it provide a valuable insight about the toxicity and about the effects of the candidate compound. Along with that, as you know, there are. It's important to know about the potential, the side effect of the compound and how it's going to affect all other organs in the body. So there have been in recent past a huge and significant kind of research to better understand the pathway activations or inhibitions, and it's being achieved by the biomarkers. So if we use some biomarkers, so it will indicate how the candidate molecule acted on some specific like organ, for example, in kidneys or on the liver. So biomarkers nowadays are playing a significant role in this all kind of the scenario, from the discovery to clinical trials and post-launch phase. As you see, as I was saying, the three main stages of the drug development, from discovery to the clinical phase to post-launch. So if you see that starting from the discovery, so there is a huge information, and the data information is generated. So for example, in the discovery phase, so there is a huge information is available from omic sciences and from the toxicology. And in the clinical phase, so there is the data from all phases, and also the clinical kind of presentations and the patient's kind of records are available. And also after the post-launch, healthcare information and the patient's form and adverse effects, the kind of records are, all these informations are available. But if we speak about the translational medicine approach, our approach is that these huge kind of information they should be a huge kind of kind of the data. It should be mined effectively in order, for example, if we have noticed some kind of side effects after post-launch, so that information should be transferred back into the bench and the bedside in order to make the new hypothesis. So here, for example, if a drug fails, still it is the variable for the future, prospective, and to make the new kind of hypothesis, it's very important. But still, a lot of, work have to be done in this phase, and to make these all information, valuable and to contribute to accelerate the all phases of the clinical trials and to reduce the time span. So overall aim is to reduce the time span and to make more kind of efficacy of this process. And so it means, so there are two fields are important for this kind of hypothesis generation and to communicate better and to extract the key points. The one is epidemiology, clinical epidemiology. So we will have the professor is going to speak about the epidemiology, so you will have a more insight. And the second one is the data management. Effective data mining and data management is the key, especially in the era of omics sciences. So a lot of approaches are being used, and one of the main important approach is by the clinical bioinformatics. Unfortunately, we also have professor, and he's going to speak about that, along with other like application of the clinical bioinformatics. So it's also being contributing to help all these kind of the problems and to help in all these the phases. So I was speaking about the data management. As I don't have so much time, so I'm going to skip the few slides. So these are the example of the a cooperation of the academia and the industry. So they have made data warehouses, for example, the Merck and the Motif Institute, especially for the oncology clinical trials. It's really a good kind of a platform, effective, and it's using various kinds of the approaches and to mine the data available. Okay. So as I was saying, and that the regulatory agencies and also industry is very much having troubles about the new approvals of the drug. So to cope that kind of situation, another kind of strategy is being used in translational medicine. And FDA is motivated to help in this aspect as well. So there are few examples in which the already approved the drugs they are being studied to see their efficacy for any other the clinical conditions, because it's already has passed the regulatory requirements, so it's easier to be approved again for the various other conditions. So one example is the muscle relaxant. As you know, so it's being is used by in the models of the addiction of alcohol to see if it is have some effect to cut the drinking habit. So the data shows that it proved excellent the results in decreasing the alcohol consumption in the rat models of the heavy drinking. So the further investigations are going on, and it can be a new kind of the repurposed kind of the medicine is going to be available. And another example of the nicotine addiction. It was studied in the experiments in the labs that to see if it can help in the reduction of the smoking habit or not. So the results in the Gallo clinic showed excellent kind of the data. It indicated that only one dose alone cut smoking in about half of the models. So these kind of findings are very promising, and so there is a huge interest from regulatory agencies as well as from NIH and other regulatory agencies like EMA in this strategy. So it's an important approach being used in translational medicine. Another important expertise is the toxicology, as you know, about the pharmacovigilance and the toxicology of so you have to be starting from the molecule if it will make the journey to the clinical trials. So to give an example of this, the toxicology, how it's important, as you know, the hepatic injury, the toxicity induced by the molecules, it's the major adverse side effect, and it's the reason of the termination or the rejection of the applications by the regulatory agencies like FDA and by EMA. So the research has been conducted at the Institute for the Drug Safety Sciences and the Hammer Institute for Health Sciences, and so they used kind of the disciplinary approach, translational medicine approach, consisting of the mechanistic, mathematical, and the biological models of the drug-induced injury in the mice, rat, humans, and the dogs. So the ultimate the product they have constructed is the DILISIM. It's a model, and according to their data, it shows that it's capable of evaluating the human toxicity of the newly the developed compounds, and by doing so, it's going to improve the human risk assessment and to reduce the drug development timeline. Of course, if it's going to tell us at an initial stage that the molecule is safe or it has huge the toxicity, so there is no need to take it to the pathway of the clinical trials. So it's an excellent example of kind of the disciplinary approach in the field of the drug R&D process. So there is another example, so where translational medicine the team was working. So in the Pittsburgh, the Clinical and Translational Science Center, they have already shown an excellent example where the neurologists, surgeons, biologists, and the bioengineering professionals and the physicians, they combined and they make a team, and supported by the CT assays. They are the funded kind of centers by NIH on the translational medicine. So they achieve an excellent kind of the finding or an achievement to restore the functional aspects in the paralyzed patients. So the idea was to, for example, if a patient is paralyzed by the stroke, so they implanted a chip in the brain of that patient, and so by just the thinking of the moving the arm, that patient was able to move his arm, and view with some exercises, even the patient was able to grab a glass of water. So it's kind of like a kind of the amazing kind of applications if we act in a if we combine and close together the various fields and the disciplines under the umbrella of translational medicine. So the such things are possible. As you can see, it provide a hope for as you see, the millions of people are affected by the stroke every year, and also the patients that are affected by the roadside accident and they have amputated like arms and the legs. So it's a good news also for those people. Important aspects about the collaboration is that academia, and they usually have a good kind of the professional skills, but they don't have a good infrastructure or instrumentation, which the industry already have. So they lack the professional expertise. So if we can combine both academia and industry, so it can be an excellent kind of team, and it can be of course, it can be a source of the potential, the revenue generations. Though there are some kind of hazards and some kind of the hurdles in doing that, especially like the problem of intellectual property and the loss and the profit sharing. But still, so there is increasing like trend to involve both academia and industry in the partnerships and collaborations. And in the TM, it's very important to involve industry along with academia, because all you are going to do in the bench or in the clinics is ultimately should go to industry, so that it can come either in the form of a diagnostic test, or it should come in the form of a drug, or like a useful kind of thing that is going to improve the healthcare. So if you are going to avoid the industry, so you are not going to achieve your ultimate aim to improve the global healthcare. So also to achieve this, involvement of the organizations, associations, NGOs is very important, as these organizations are going to provide you with an alternate source of the funding and access to the patients, groups that you can also use to enroll the patient in your clinical trials. And it also provides a good way to enroll the volunteers. And so these are some kind of the strategies and approaches. So these are more or less, they are non-scientific, but these are important to have a good infrastructure of translational medicine. So you can just, keep them in mind that a good, project, the manager, it can be a much, the useful to do a better communication between the bench side, the bedside, and along with the regulatory expert, it can push up your things related to the patents and the licensing stuff, and take care of your grant applications. So these are the points that are not even gained importance even in academia by the PIs, principal investigators, and professors. But so they cost like a normal postdoc, but there the benefits are huge, and they can contribute huge if you are going to employ them and to take care of your kind of communication and your applications. And if you have some on-site the facilities in the form of the research lab or some instrumentation, so you can ideally you can earn an alternate source of income I mean, by renting, especially in USA, but it's also possible in Europe. So these are some approaches to have some alternative source of the funding to keep the translational medicine infrastructure, I mean, going. So a brief overview of the global initiatives and the programs. If we speak about the USA, about the 2 years before, NCAT was established by NIH. The aim and mission of this NCAT was to reduce and remove and bypass costly and time-consuming bottleneck in the translational research pipeline in an effort to speed up the process of the new drugs, diagnostic, and the medical instrumentation to the patients, and to overcome all the previously started translational medicine programs under the umbrella of NIH. So NCAT is doing a significant contribution in the recent years. So for example, so they are making tools to promote the common standards and the best practices, especially in regard to the patient recruitments and the consenting. So they are making a harmonization and like a standardization all over the USA. And also they are making a standardized kind of procedure for the data capture and the study management protocols. And to strengthen the efforts, so they have make partnership with the regulatory agencies like FDA and the DARPA. And especially with the DARPA, so they are having the projects to develop the tissue chips to speed up the toxicology assessments. So it's kind of a huge investment has been done by the developing the drug, I mean toxicity to the chips assessments, and the various organizations are involved in USA. So the CAN is also the contributing and helping the NCATs by providing funding for various kind of initiatives. As I was speaking about the repurposing the drugs and the tissue chips and identification and validation of the drug target. So in all these kind of the research projects, the CAN is providing funding to the NCATs. So in about 62 the states in USA, so I would say not like states, but the institutions. So there are 62 institution consortiums of the CTSA. So they are the clinical and translational science, the centers. So they were started by NIH in 2006. Though they made some progress to better understanding of the translational medicine and to make a move, but I mean, along with the progress, so there were some hurdles and there were some kind of the bureaucratic problems. So now these all the CTSA programs are under the umbrella of NCAT. So NCAT is going to look after all these 62 institutions consortium and to monitor their the annual progress. And these CTSAs after coming under the umbrella of the NCATs, so they have developed the various the portals of informations. For example, the Research Match, the Red Cap, CTSA IP, and Citipedia. So these are kind of the information portal where you can have an access to the databases of the of intellectual property or the patent informations, and about the kind of the data on the various aspects of the clinical conditions. And if you want to have access to some the regulatory agencies in other part of the world, so it's also possible. So under the umbrella of the NCAD, CTSAs are making a fast approach to achieve these all kind of the milestone they have been set by the NCADs for annual from annual to annual year inspection. So if we speak about the changes in the CTSA program, so they have NCAT has now started a single institutional the review board model, and so it's going to reduce the bureaucratic and administrative stuffs, and so they are developing effective data collection and the mining tools. It's also going to provide a standardization to all in all states, and so they are already actively making the strategic alliances with other organizations. And along with that, now they have realized that the community involvement is very important. So they are actively in this area. They are active also to make the process of the patient recruitment. So these are kind of the changes that has been done after the NCAD the takeover the CTSAs. So another example that so by the CTSA, so there was the first newborn a screening study was held of the Fragile X syndrome at the Davis CTSA Center. So it was not the possible before to do this in the newborn the screening study of the Fragile X syndrome. So it's also like kind of a teamwork. As I don't have time, so I would just skip. And in the European Union, so we have here Horizon 2020 program. So it's the funding tool of European Union. So there is a more kind of emphasis for in the key instrumentation and the technologies, and there is a great amount of support for SMEs companies. And the one major the point in this program is that they have started new strategies and approaches for international cooperation. So it's a good news for those are not based in Europe. So United Kingdom have a cutting edge, a good infrastructure of translational medicine. So NIHR, they have funded 5 comprehensive basic research centers that deal with the broad spectrum of the topics, and 7 specialized centers. So they revolve around the one specific area, such as the pediatrics. So these kind of specialized, focused centers, they have contributed significantly for TM infrastructure or the scenario in the United Kingdom, and it has resulted in a significant increase in the number of the clinical trials in UK. So if we go to the Singapore, so the Singapore is going to be to lead translational medicine in the Asian region. So there are the various initiatives and activities that are going on in Singapore. They are attracting the Western professionals at a very high incentive, incentives and other kind of stuff to recruit and to help Singapore to establish the infrastructures there. So there is the famous A-Star Institutes, so they are working there. And so there is a Singapore Institute for the Clinical Sciences, and so there is a cooperation between Duke and the University of Singapore. And there is an academy of the GXP excellence. So all these kind of the initiatives are the significantly, contributing for making Singapore a leading country in the Asian market. In India, there is no significant translation medicine infrastructure, unfortunately, but there is increasing awareness. So as a result, so Harvard, MIT, HST, and the government of India have started a partnership to start a translational health science and technology institute. So it means that it's going to be, so there's increased awareness, so, but it will still take some time that India is going to be in the translational medicine field. In Japan, the government is very much committed to achieve the aim of translational medicine. So they have assigned some biopharmaceutical targets to each individual universities that they should focus on them specific kind of areas and research. Along with that, they are providing already integrated translational research, the training in the medical schools. And there are some specialized institutions, for example, the Institute for the Frontier Medical Sciences. So it's very active in translational stem cell research. In Australia, NHMRC is the main funding body. So they have already invested huge to build the translational medicine infrastructure in all over the country. So if you go to the universities in Australia, you will find the various translational medicine forms, and the master programs are in many the universities there. And with the huge investments by the government, so they have established a translational research institute in Brisbane, and it's going to play a very active role in the field. In some the part of the like Australia, so they are providing the translational medicine framework based on the three main pillars: research, education, and clinical care under a single roof. And one example is of this institution is the Macquarie University Hospital, where under one roof you have the research, education, and the clinical care. So in China is also aware of translational medicine, and along with Singapore, they are also struggling, and they are going to start or like to establish infrastructure for translational medicine. So you can see that from start of the 2009, so they are already establishing translational medicine institutions in various the university hospitals and some postgraduate institutions. Don't know how much time I have. Anna? Okay, so briefly about the translational medicine education and trainings. So there are two main aspects. So we have to educate the current professionals that have background in pharmacy, that have background in the veterinary, and that have background in like medicine or the molecular biologist. And the second aspect is that we have to produce the future translators. So if we will speak about to educate the current professionals, so it can be achieved by providing individualized, the tailored programs, seminars, symposia, workshops, and to provide the professional the certification programs. If we speak about the future, translators, so an ideal translator should have the better understanding of the human, medicine principles, the better understanding of the pathophysiology, and along with that, basic and human pharmacology. So it's not enough, but along with that, he should have also a better knowledge of the clinical trials, the regulatory affairs, and the business matters for the industry. So all these kind of the skills are important for a future like translator. And if you will see the curricula of the most of the universities all over the world, so so there are some programs that have all these stuff, but most of them they have like some part. So they have focused on some parts and not all. So there are the kind of the few aspects of the translational medicine education and training. I have mentioned the few like kind of the fields so that are important and they have the high demand in industry. And also the training aspect of that is that there should be rotation in the clinics, industry internships, and research stay abroad, and involvement of the pharma and biotech industry as a program co-sponsors. So it will end to create advanced job placement and the comprehensive the medical translators in the market. Just the brief introduction of the USTM. So we are a non-profit, the neutral platform for the advancement and progress of TM. So our overall aim is to enhance R&D process of novel and affordable diagnostic tools and the treatment of clinical condition affecting the population. So here is the website address. So we have annual conferences every year. So it's going to be held the next month in Vienna in Schönbrunn Palace. So if any one of you want to join, you are most welcome, and to get more insight about the translational medicine. So we are going to have the stakeholders and key opinion, the leaders from over like 40 countries, including speakers from FDA, EMA, and NASA, and other organizations. So we are going to have scientific and panel talks, exhibitions, and so on. So we have the Global Translational Medicine Consortium. It consists of the members include the research groups, pharma industries, CROs, small and medium enterprises, and the NPOs. So we have nowadays about more than 40 members from all over the world. And to provide the professional, the certification in translational medicine field, so we have Academy of Translational Medicine Professional, and under this umbrella of Academy, so we are offering professional certification programs in biomarker, in clinical bioinformatics, in public health as well. So you can just explore the website if you are interested. And we are having our inauguration of our the scientific publication of the USTM New Horizon Translational Medicine in September, the next month. And so in summary, translational medicine has the great potential of accelerating the R&D process. So there are already multiple global initiatives and programs are already working. But need is to standardize TM training and educational activities and to optimize the TM tools and the techniques. So it makes the end of my presentation. I would like to thank you for the listening the talk, and if you have any question, you can briefly ask as we don't have so much time. So after my talk, I would like to invite our next speaker, um, uh, the Professor Raquel Lucas from the University of Porto. She is expert in the public health and epidemiology field, and she is currently working in the University of Porto. And she is the lecturer in epidemiology, and the research interests are etiological epidemiology of the musculoskeletal conditions and the trends and the predictor of HIV incidence at most at-risk population. As epidemiology is contributing a very important role in nowadays the clinical practice, so it would be very interesting talk. So I would like you to welcome Professor Raquel Lucas.
Good morning, everyone. First of all, I'm sorry, but actually Sarah had told me that I would have to give her my presentation ahead of time, but I just took a little bit more preparing the slides. First of all, I have to say that I'm very honored to be here to speak to this large crowd of pharmacy students, because I'm also a pharmacist, and so it's very interesting for me to see how things have evolved since I graduated, which was some 10 years ago, so that's a long time really for you. So I'm going to try to speak to you about how we do epidemiologic research and some of the challenges that we face nowadays when we try to answer public health challenges. So the thing with public health is that we're trying to prevent disease. So most of the times we are interested that nothing actually happens. And it's very difficult to measure nothing happening. So when actually something happens, that's when we have to use epidemiology. Us epidemiologists would like everybody to know who this man was. He was Jon Snow. I don't believe that it's very likely that you've heard of him. Has anybody heard of Jon Snow ever? Okay, great. Oh, that's good. Because Jon Snow was a doctor. He was an anesthesiologist who lived in the 19th century. And for those of you who don't know about him, he was involved in studying a cholera epidemic that happened in 1854 in London. And the thing he did was, he was the first person who was considered the father of epidemiology, because he was the first person to actually map the occurrence of disease and to establish the transmission route for a disease without knowing the infectious agent. So this was before Vibrio cholerae was identified. And what he hypothesized was that cholera was transmitted through water supplies. And the opposing theory at the moment was that it was miasmas who transmitted the disease. So it was contaminated masses of air. And John Snow was convinced that water was the actual problem. Remember, nothing about the bacteria was known. So he actually, well, he identified the problem, that there was a huge epidemic of cholera in the central London. And then he decided to map the deaths by house using a map of London at the time. And so by doing that, he understood that most cases were near a water pump, which was probably the responsible for transmitting the disease. So he actually was quantifying a difference between different water supplies and different death rates. And then he inferred causation, which is what we want to do by using epidemiology. We want to infer that by a statistical association, we can prove or disprove causation. And then he went on to a preventive action, which we do not think was the main, the main outcome of the epidemic, since the epidemic was already decreasing at the time, but actually the handle of the pump that you see over there was removed. And so that was a very symbolic gesture that meant that health authorities recognized that his theory was right. And they also didn't do a formal impact assessment. So nowadays we do need that. But at the time, history ended up proving that John Snow was right. And so that was the impact assessment. At least he had a very huge impact in the birth of epidemiology as a discipline. And since then, epidemiology has evolved a lot. So in the 20th century, we went from this map by John Snow, which told us that actually the interactions between the human host, the environment, and an infectious agent were very likely to produce disease. But then we went on to the chronic disease epidemiology. So the chronic diseases became the most important burden of disease in the most developed countries. So it was through epidemiologic studies, through comparing groups within populations, that we understood that cigarettes were bad for your health, that smokers had a higher mortality and higher burden of disease than non-smokers, that we learned a lot from the Framingham study on the classical risk factors for cardiovascular disease that we use nowadays and that are renewed every couple of years. And that we have also learned a lot about the interactions between gene exposures and environment exposures in producing disease. And that has to do with, well, this here is the example of the factor V of Leiden and thrombosis interaction with oral contraceptives. But at this moment, we have increasingly complex challenges. Everybody recognizes this ecological model. Everybody has seen this in one way or another, in the sense that we think of health now as a product of a huge amount of interactions that act and interact between themselves at several levels in order to produce the health of the individual and therefore the population. And this goes from the tiniest, the most fundamental pathophysiological pathways until the actual cultural and political organization of society. And so, and that happens within an environment and also within the life course. And this is a key aspect. But the thing is that many times we focus only on one of these levels, even though we understand that the whole model is the important thing. So we now see epidemiology, and this is to distinguish it from lab science, as the study of the ecology of disease. And when we are studying the ecology of disease, we have to start by characterizing who has the disease, what's the distribution of disease. So we start by seeing those maps. Everybody also has seen this map or a similar kind of map. This is infant mortality rates by country in the world. And it's not surprising because we have seen this message a lot. This has a lot to do with human development. And actually, we do see that there's a disproportionate burden of disease in sub-Saharan countries, actually in the whole southern hemisphere. But this is not just a description. We can see this as a map which describes a problem, but we are actually trying to explain the problem also by presenting things geographically. And we all have hypotheses for that. Some are proven, some are yet to prove, but actually exploring disease distribution is never just exploring disease distribution, and we'll see that ahead. Also, it's not enough to describe geographically how things evolve. We have to understand them throughout time. And, well, you probably cannot read all those small print, but those are regions of the world. And on the x-axis, you see the mean age of death in 2010, and on the y-axis, you see mean age of death in 1970. So what we see here was that if death ages were kept constant in all countries would be in that line where x equals y. And what we see is that in every region of the world there was improvement in mean age of death. So people are living longer everywhere. Of course, it's not symmetrical. Not all the countries have evolved in the same way. Not all the regions are improving. But this is something that we have to keep in mind, that data can be analyzed in many ways. And actually, this is much less seen than the map that you saw before. And us, at the moment, we are here. It's great. I mean, we are improving and we are living a lot, but we can also zoom in on the distribution of life expectancy. And what you see here is that even if we zoom in, inequalities appear. So in the previous world map where we saw that all the European region had the same color in terms of infant death rates, now we see a completely different picture. And you see that there are some countries that are in the highest life expectancy quintiles, but for instance, Portugal, where we are now, is not. And we might all have explanations for that too. And this suggests that a lot of research has to be made. And it also suggests that we have to face things at a macro level, not just at the tiniest level possible. Well, that happens when we are comparing different countries in the European region, but this is just a city. This is Washington, D.C., and we're talking about disparities in health here again. So this is life expectancy in different regions of Washington, D.C. And what you see here, this is, all these 72s are the life expectancy in the city center, in Washington, D.C. center. And what you see in the periphery are the life expectancies of the regions, of the suburbs, where the metro lines end, actually. So beyond actually comparing countries, we can even find inequalities when we are speaking about much shorter distances. So here we see that there's almost a 10-year difference between in life expectancy between the city center in DC and this northern suburb. So we have to actually explore that, and we're not only interested in improving countries as a whole, but even within the countries there are much more subtle differences that sometimes are not clearly measured. So that was the distribution, and how do we prove that something causes that distribution, because that's what we're interested in. We use statistical associations. And that's the most interesting thing, and at the same time, the biggest problem with epidemiologic research is that we have to rely on statistical associations. We cannot use lab experiments where we manipulate all the conditions except the exposure that we're studying. So this is what happens when we use aggregate data. This is a graph where you see a lot of countries, and then you see that the x-axis is income inequality, so how much are the richer than the poorer, and on the y-axis you see the life expectancy. And of course, this is made for us to conclude that actually income inequality causes a decrease in life expectancy. And this can be true, but it can just not be true. It's very difficult to infer causation from these kinds of aggregate data because we're talking about ecological correlations. And we do have a problem because we do not know if actually the people who are poorer are the ones who have the lowest life expectancy. Of course, we have by now a very reasonable amount of evidence that tell us that there's actually a causal relationship between these two, but in terms of the production of knowledge, we have some limitations with inferring causation from this. And this is this is a kind of a joke that is based on ecological data, because we are seeing more and more of these correlations between some variable and another health outcome in terms of countries using aggregate data. And this is actually, on the x-axis you see chocolate consumption per capita, and on the y-axis you see the number of Nobel Prizes. And actually the correlation coefficient of the line that you can use to describe this association is really high. It's like it's almost 0.80, and that's a high correlation coefficient. So if you want to infer causation, you can do it by this. Only, what's the difference between a well-performed study and some association that we randomly found because we are actually asking the wrong question? So what we want when we are conducting epidemiologic research, and let's go back to the reason why we compare groups of people. We have a target population, and we want to find out the effect of some exposure on some outcome. So ideally, what we would do is we would expose all the population to our factor that we're studying, and then we would unexpose everybody at the same time, keeping all other things constant, which is impossible, of course. Only philosophically can we do that. So in practice, what we end up doing is to find statistical associations. We compare a fraction of people who are unexposed and the fraction of people who are exposed with regard to the frequency of the outcome. And if there is a statistical association between exposure and outcome, then we try to infer causation from that statistical association. And the problem of this is that in randomized controlled trials, as you know, we can allocate randomly the exposure. So the white group and the blue group are actually exchangeable in terms of all characteristics, the ones that we can measure and the ones that we cannot measure and which are potentially even more important. So if they're exchangeable, all the effect that we see has to do with the exposure that we are providing those people. But the thing is, because of ethical reasons and practical reasons, unfortunately, really, we cannot expose people to what we want to causative factors of disease. And thankfully, so what happens is that we conduct most of the time observational studies, and those observational studies are studies where we compare two groups, one which is exposed naturally, spontaneously to the factor that we're assessing, and the other one which is not exposed to the factor that we're assessing. And then we compare them again with regard to the frequency of disease. But the question is, are those groups exchangeable? Will the unexposed group be the same as the exposed group had they been exposed? So now we are talking about bias and confounding. Bias has to do with the way that we select people for studying, the way that we collect information from those people. Confounding has to do with the presence of other factors that are actually causal factors which explain the statistical association that we see between an exposure and an outcome. So what we want is those groups to be exchangeable. And that's why people came up with these hierarchies of evidence, those evidence pyramid. Everybody has seen this in one way or another. And there's one interesting thing is that they're all different. Everybody does their own evidence pyramid. But there is actually something in common between them, is that experimental studies are always on the top of the evidence in terms of the quality of evidence that they provide. Then we see cohort studies, observational studies where people are followed up throughout time, and then case control studies usually, and then cross-sectional studies and case reports and case series. Why is this important? I don't think there's a universal classification of the hierarchy of evidence, and that's why I brought you all these pyramids here. However, we do have to bear in mind that this ranking of the types of studies has to do with the probability that we can eliminate bias and confounding. So the probability that each kind of study here will give us a valid result, a result which actually represents the effect that we want to measure. And this is an example of why this is important. And this is an old example. It's an old example because you're very young, and I think most of you know this example, but probably you don't, you weren't studying at the time that this problem started. I was actually entering college at the time. So what happened was there was this doctor which had a pediatrics clinic and who observed 12 cases of children who had an autism spectrum disorder, and whose exposure, identified by either their parents or their doctor, was the measles, mumps, rubella vaccine, the MMR vaccine. And so, after observing this association, something that he thought, the whole team thought was a potential association, they actually started posing the hypothesis that actually the vaccine was causing these cognitive development disorders, and that was a huge problem. That was a problem because, well, it's a vaccine which has actually universal coverage, as you know. And so it's also a preventive intervention. So you give it to people who are not sick, so they don't need it. And there's an additional thing, it's administered to children. So everybody, all children, and they're not sick. So that's a problem in terms of the society, and it should be a problem. And as you know, then started, as you might know, then there was this, in the beginning of the 2000s, there was this huge discussion in the public, in the lay media, about whether the vaccines would cause autism or not. So that was on the basis of that paper, which had 12 patients and no comparison group and no adjustment whatsoever. However, you see that this actually reached epidemic proportions, the scare about the MMR vaccine, and it still persists today. This is a news that was, well, it was about Tony Blair's, who was the British Prime Minister's son, and that there was actually a lot of discussion in the UK about him having vaccinated his son or not. And in the end, that's actually irrelevant. The problem was that when people actually were able to do studies which were observational, but which had huge sample size. This is 2 million person years, so this is like following 2 million people for 1 year. They're all children. And this was done with Danish data, where they have this very complete registration system for health issues and for other issues also. And what they saw was that regardless of the age at vaccination, regardless of the intervals since vaccination, and regardless of the calendar period of vaccination, there was absolutely no association between being vaccinated, being immunized with the MMR vaccine, and showing autistic disorders or other autistic spectrum disorders. And this is very important. However, negative findings are very difficult to manage in terms, are not very sellable in terms of communication. This was a very robust study, and along with a huge amount of other evidence, it led to the retraction of the paper, the 98 paper, with the 12 cases, because actually there were several methodological flaws. But the retraction happened 12 years later, so that's important. Bottom line, well, there was this study. It was not very strong because it didn't have a comparison group, and then there was a huge amount of studies that refuted the hypothesis that the first study put. However, what happened was that in the white bars, you see the MMR vaccine coverage, and the blue line, the dark blue line, is the number of measles cases in the UK. And this is very, very relevant because, as you see, as the coverage of the vaccine decreases, we go below the threshold for herd immunity. And that for measles is very high. So you need more than 90% of children to be immunized in order to obtain herd immunity, which is no transmission of the disease in the population. And what happened is that, well, we thought measles was controlled, but it wasn't in the end, because coverage decreased below that threshold, and then the number of measles cases in the UK increased substantially. And that was an unpredictable problem in terms of public health because we actually had the best knowledge. So there was actually no scientific reason for this to happen, which leads us to the importance of maintaining critical overlook over these the kinds of studies that we can have, and of the importance of actually improving methods for causal inference when we are working in epidemiologic studies, and that's particularly important for pharmacoepidemiology, really. And that's an area which it's probably more related to your future field of work, really. Besides describing where the disease is, we have to, and what the intervention is, if the intervention is effective or not, we have to define the target population. And in terms of public health, we can act on all these stages of the disease course. So we can modify prognosis, we can improve the uptake of diagnostic techniques, we can actually try and anticipate diagnosis by using screening. But in terms of the impact that we will have on the population, we would like to work or to intervene in that phase. So the sooner we intervene, the better is our chances to prevent the diseases from occurring. So in terms of public health, we want to intervene as early as possible. And what's as early as possible? Well, it starts before birth, actually. We now know that many chronic diseases are the result of either an accumulation of exposures or some critical exposures that occur either in utero or in the first years of life. And those actually work together to produce or to increase or to modify the risk of a disease throughout adult life. So it's not just about exposures during adult life, it's the whole life course and the whole accumulation that counts. This is an example of how that can work. And we don't know actually a lot about how that works in practice because we haven't been able to follow people as long as a life course. However, there is a lot of evidence that suggests that, for instance, we have, if you see the solid line in the graph, that's the full genetic potential for acquiring bone mass. And the dashed line is what you get if you have inadequate environmental contexts. So if you don't have enough nutrients or enough physical activity, you don't reach your full genetic potential. So that means that the fracture risk in old age, the fragility or osteoporotic fracture risk in old age, is actually a function of what you acquire during the first decades of life in terms of the bone mass, really. So this whole reasoning of optimizing the characteristics or optimizing actually health during the first decades of life, during the first years of life, was what led to the establishment of birth cohorts. Birth cohorts are actually groups of people who are followed up from birth until, desirably, until adult life. And we are starting to get a lot of evidence on the causation of chronic diseases by using those birth cohorts. Actually, what I'm bringing here is a birth cohort in Porto that, whose research team I'm a part of. This is Generation 21. Generation 21 is a group of over 8,000 children who are followed up throughout time. And so they were born in 2005, 2006, and now they're 7-year-old children. And what we are trying to understand is whether characteristics of birth are actually predictive of what we are going to observe later in childhood and also in adult life. And we are particularly focusing on body composition and how obesity tracks throughout childhood and into adulthood, and also whether there is a transgenerationality in completely different constructs, like, for instance, physical abuse, which is, of course, very important. And it's an important determinant of, not only physical, but psychological and psychosocial health. So this is the future, starts by studying the determinants of disease as upstream as possible, really. Well, besides knowing when to study people and how to intervene on them, we have to know if what we propose to the population actually works. So we have a number of studies on efficacy, for instance, of other medical interventions. But there's actually a gap between what we think works and what actually works in the real world. These are standardized mortality rates from coronary heart disease. And I wanted to bring you this because in most countries, coronary heart disease death rates have decreased substantially in the past decades. And there are lots of explanations. Well, there are two main possible explanations. One is that we were successfully able to act on risk factors, and one is that the treatments are getting better. So either people are getting less disease or people with disease are surviving longer. And this is, well, these are the main possible determinants of those changes, those trends throughout time. And this has been used in the impact model. The impact model is a model which uses data from countries, aggregated data from countries, in order to assess whether to which extent we can use risk factors or treatment to explain trends in coronary heart disease mortality. So what we see here is that we use a base population distributed by age and sex, and then a number of risk factors that we know that are important, like cholesterol, BMI, blood pressure, diabetes, smoking, and physical inactivity. It means we have to have data on those. We also have to have data on the treatments for acute myocardial infarction, heart failure, and angina. And that means pharmacological and surgical treatments also. And of course, we have to have the outcome, which are, in this case, coronary heart disease deaths. And what a group of colleagues of mine did was, well, first they predicted the death rates that would have happened if the mortality rate by coronary heart disease remained the same from 95 to 2008. And they observed that actually there was a decrease in the number of coronary heart disease deaths. And this could be because risk factors are better or treatments are better. And what they did was they used, to model the deaths for each cause, they multiplied the number of eligible patients, and for instance, in the case of pharmacological treatment, by treatment uptake, by case fatality rate, and relative mortality reduction. And so what they got in the end was something like this. They they were able to pinpoint, they were able to attribute the whole decrease, the whole trends, the whole decreasing trends to each of these risk factors. So what we see is that by a fraction, risk factors worsened, in particular diabetes in men and women, and BMI in men and women. That's the Portuguese context. But we also had a number of risk factors that improved a lot, and they improved more than the other ones decreased. So. Actually, we saw that for a number, like systolic blood pressure was the most important, and total cholesterol, there was an improvement in the overall the population. But we also saw that treatments were very helpful in decreasing the mortality by coronary heart disease. And what does this, what does this boil up to? What we see was that the trends that we have observed were explained more by treatments than by risk factors when you have the whole model together. And this means that we are being very effective in treating people, and that we are being comparatively less effective in preventing the uptake of risk behaviors and risk factors overall. So that has, of course, important implications in terms of public health, which means that we are actually moving towards a more medicalized society. That's not true for all countries, and here we see differences, substantial differences between countries. Like in Iceland and Northern Ireland, prevention was much more important than treatment. But in many countries, well, the contributions are actually very similar. And why can we not act as well as in a perfect way? Why can we not, well, give everybody the conditions for them to have, well, decrease their risk factors and uptake treatment? There are a number of reasons, but now we have to look again for groups of people who are in particular risk of, for instance, not adhering to a treatment. This is what we have observed, what a colleague of mine has observed in terms of dyslipidemia treatment. Cholesterol lowering therapy is much more likely to be needed by men. However, men are much less likely to attain the therapeutic goals for cholesterol reduction. So this is clearly identifiable as an asymmetry. So even though we provide treatment for everybody, they are less likely to succeed in treatment. As you know, there are many differences between given groups of people and the way that they are successfully treated. And this is also an important inequality. This is the proportion of people who leave the hospital, who are discharged with the adequate treatment for an acute coronary syndrome. And so we would expect that if there are no contraindications, everybody, regardless of their age, would have the same treatment. However, what we observe is that there's a relative divestment in people with older ages, even though there is no actually clinical reason not to treat people with older ages. But actually, we do see that the best practice is not always the actual practice. We also see that there is a number of health commodities that are provided only to people with well, an educational background or a social background which is more well off, really. And this is calcium and vitamin D supplementation among women who are doing osteoporotic therapy. And we would expect that all women had access to supplementation with calcium and vitamin D, given that they are doing anti-resorptive therapy, but that doesn't happen. And we actually see that as the more educated women are, the more likely it is that they are actually doing calcium and vitamin supplementation, which is recommended by the guidelines. But we also have to have a critical view on what we are delivering to people. This is the rates of C-section in births in some European countries. And again, this is very interesting for the Portuguese context in particular, because we have a very high rate of C-section. And there is actually no, no rational reason why we should have a very high rates of C-sections in Portuguese births, because there is actually no indication for that. And we see that happens a lot more in the private sector than in the public sector. And it's something that happens in a lot of countries. It's something that's characterized. And in this case, we are actually providing people a technology that they don't need. So a very complex surgical procedure that is actually putting them in a disadvantages in terms of women's health and in terms of the child's health also. But we also have to bear in mind that it's not only the provider's problem. It's not the providers that modulate the risk of C-section. It's also the women themselves, because there are actually cultural factors that interact, or that women have that interact with healthcare provision, and that, for instance, in Portugal, we know that women in the exact same clinical condition, when they enter the maternities, if they're Brazilian, they're more likely to have a C-section than Portuguese women. And that's all in public hospitals. So we do have to bear in mind that cultural background is very important in the way people manage their own health, in the way that they interact with the health system and with the health professionals. Again, for preventing disease, people have to actually adopt prevention behaviors. This is the case of HIV in Portugal. These are reported cases of HIV in Portugal. These are the most recent data, really. And you see that what I would like to underline there is that Injecting drug use, which is the gray line over there, has been decreasing a lot. So after some decades where IDU was the main transmission mode for HIV in Portugal, now we actually have seen an increase in an overall increase in the number of notifications of reports of HIV infection that are due to sex between men. So men who have sex with men are now the most worrying group in terms of HIV transmission in Portugal. And don't be fooled by that heterosexual transmission being on the top, because, well, if you divide it by the total population, the risk is much lower than it is among MSM. And why does that happen? Well, because people are actually getting tired of primary prevention. So we cannot, as health professionals, we cannot be judgmental. We cannot think that people, well, should use primary prevention, and to blame their health state blame themselves or their choice of behaviors for their health state. So that's very judgmental, and that's not what the health professionals should do. We know that people have sex with many partners. We know that people have unprotected intercourse. And we know that they shouldn't have unprotected intercourse. And they still do. So that's a problem that we have to solve. And it's probably not just by educating people, really. We also know that people use drugs, people inject drugs. And that's a particular problem when people use drugs during or before intercourse. And both are together, that's a particularly important problem. However, they get tested a lot. These are only men who have sex with men. These are the results of a survey that we conducted among MSM, a large survey. And what you see is that people, actually, more than a quarter of all the MSM that were questioned had one or more HIV tests before. So people are aware, MSM are aware that actually they have the risk of contracting the disease, contracting the infection. And so they use testing as prevention. That's secondary prevention, early testing. And of course, that is questionable because it does mean that we are in the face of people choosing to be at a higher risk than they would need to be. We conducted a study. Well, this is a board that I saw in New Mexico, in Albuquerque, that had all these kinds of posters of flamenco classes and yoga classes and all. And in the middle of that, there was this pamphlet with free rapid HIV testing. So that's the total normalization of HIV testing. Like if you go to, it's either you go to a flamenco class or you go to get to HIV testing. And that means a lot in terms of how the society is changing. And that's why we took advantage of the fact that there is a center for testing, HIV testing for men who have sex with men. It's in Lisbon, and where they provide free anonymous testing, and where men who have sex with men get tested by other MSM. And the objective is to provide, well, testing to anticipate diagnosis, and also counseling on how to decrease risk behaviors. So we found out that at the baseline screening, 4% of people were actually positive for HIV already. But that's not the most important thing. They do a rapid test, and for those who are negative at the baseline screening, we offer them follow-up. So they get tested. The peers there, the technicians, test them periodically, like every 6 months or every year. And we, as the researchers, are able to measure incidence rates of infection, which is much more interesting and much more dynamic than actually screening for prevalence. We want to know how things change, how things evolve, if people are getting more infected or not. And these are preliminary data that we have obtained from that. And what this means is that if we follow 100 men for 1 year, 3 to 4 of them will get infected by HIV. that's a group of men who have sex with men. Of course, this is not representative of the whole gay population, but it's worrying nevertheless. This is, and this is worrying not just because it's a high number of seroconversions, but also because the determinants of that are, well, it's very interesting to look at this, because we would think that the most important determinant would be unprotected anal sex with an occasional partner. So people who have these risk behaviors and those behaviors that are associated with an obvious increase in risk. But what happens is that the most important determinants are the use of recreational drugs before or during sexual intercourse, and also the fact that people have an HIV positive regular partner. So this is risk management. People are actually adopting some risk behaviors with their regular partners because they simply choose to. So just to end, I'm sorry, because Sarah is... It's already, trying to kick me off the stage. So finally, what I wanted to leave you with is this message, that there are many ways to do science, and sometimes we are sure we will get valid conclusions, but sometimes we're not. There is no universal, fantastic study that will answer all questions, but even though we are in presence of imperfect evidence, we have no excuse not to act on the best evidence possible. And that's the message that I would like to leave you. So, in the end, I hope you remember that public health is actually about the organization of society and not only about the absence of disease. Thank you.
So if you have any question, I think they need coffee. So you have the 20 minutes, the coffee break, so enjoy the coffee.
Hey guys, I'm going to ask you to sit, please, to continue the scientific symposium. Okay. I'm going to kindly ask you to sit down, please, and do some silence because we have to continue, please. Let's go. Over there in the bottom of the room, please sit down. Please hurry to your places, please. Thank you.
I hope you have enjoyed the coffee and now in the remaining speakers of the morning sessions. So I'm going to introduce our next speaker. He is experts in the bioinformatics, very important field as I have highlighted in my talk. And the cutting edge approaches, so using the various expertises in bioinformatics. So the Professor Joel Lewis is affiliated in the Department of Electronics and the Telecommunication and Informatics at the University of Aveiro. And he is also the founder of a BMD, it's an SME company. And it's working in the biomedical informatics field. And Professor, the research interests include the information, the retrieval, text mining, and distributed system and computational methods in biomedicine. And Professor is involved in many the European Union projects. The few of them are FP7, IMI, and he's the author of more than 200 the publications and book chapters in various international peer-reviewed journals, and he is invited speaker at international conferences. So I would like to welcome Professor Joel Lewis.
Hello, good morning. I wish to start thanking the organization for this invitation. My background is a bit outside your interest. I will try to be quite general that you understand what I'm talking about. So Sorry. Okay. So the first question that I want to discuss with you is what is bioinformatics? Bioinformatics is a recent research area that grows in the confluence of molecular biology or genomics or omics science and informatics. And it's becoming quite popular and also quite important. Besides bioinformatics, there is also a more broader area that is called biomedical informatics that joins a bit bioinformatics and try to apply this, the results of bioinformatics to medicine. You already heard about translation medicine, and the translation medicine in some way is in the middle between biomedical informatics and medicine. So bioinformatics is a field with lots of challenge. And in the last 10 years, in our group, despite we are mainly a computer science group, and you may saw in the previous slide a set of buzzwords around bioinformatics and biomedical informatics concepts. We have been working on the last 10 years in trying to apply computational approach in molecular biology problems and medical problems. Here you see some research line of our group. It's quite diverse. From genome analysis, for instance, we have been interested in identifying patterns in genes that helps to explain mRNA mistranslation, for instance. We have also been working on gene synthesis, microarray analysis, and next generation sequence. A second field of research is dealing with big data that medicine, pharmacology, molecular biology every day creates nowadays. So data and knowledge integration is also a quite important issue that we have been trying to address. The third research field is related to text mining. I will concentrate most of my talk on this particular problem. And last, fourth research line is related to biomedical informatics. We have been a very applied research group, and most of the results of our research we try to deliver something to the end user, either a physician, either a researcher or a molecular biologist. So let's discuss a bit what is text mining. We all know the problem of data deluge in biomedicine. Every day we have more information available in forms of books, in forms of scientific papers, or even in social media. So the amount of data that is daily generated around the world in our particular field is amazing. So the main question that we ask ourselves, how can we read all this data? We can't. We cannot. So how can we capture the knowledge that is being put in those documents? This is a huge problem. Of course, we have technology from our side. So internet already helps a lot exchanging the data and the knowledge. So most of our learning skills today comes from social interaction with other colleagues and other professionals. We also use tools like Google. We cannot even understand how we can do research without Google. So most of the time when we need to address a specific homework, for instance, for a course, or doing research, one of the first things that we do is Google for specific concept that we need to learn more. So let's go back. What is text mining? So in here, you just see something that is already familiar to you. It's a PubMed abstract, a scientific abstract. And you already passed this problem. You need to read many documents, many papers, and to try to figure out what is inside each paper. So text mining is about using computational algorithms that read and learn for us, and also extract some information that most of the time we don't realize that are in the documents. For instance, we can see that in this abstract there is a specific assertion that say that a specific compound, say compound A, interacts with target B. And in another paper, we can also see that B is somehow associated with C. And we need to read both documents to infer that, in fact, maybe there is an association between A and C. So text mining can help to understand those connections. So in here, the goal is to identify what are the main entities or concepts that are present in this abstract, but not only the concepts, but also the associations, the events, and finally the assertions, the main facts that are the key points in this abstract. So in our group, we have been working on this challenge in text mining, and we have been working, for instance, query expansion, name identity recognition, that is the recognition, the automatic recognition of those concepts. And I will show you some of those applications. Well, maybe 5-6 years ago, one of the first tools that we developed is about query expansion. What is this stuff, query expansion? Typically, when you perform a Google query, you type a name of a protein. Query expansion means that the engine will add other terms that you don't know, but the system knows that with inclusion of those terms, the results will be more efficient than without those terms. And this is query expansion. The motivation for this tool was this. At that time, we were working on microarray analysis, gene expression, basically. And when you perform gene expression experiments, the result will be a set of genes, co-express it, upper-regulated or down-regulated. So if you want to understand what are, well, 20 genes from an expression, what are the commonalities between those genes? We need to read lots of papers again, trying to figure out, are they in the same metabolic pathway? Have they some molecular characteristics that allows to identify them in this particular condition? So we developed this tool with which the main entry point is indeed a set of gene IDs. We expand those gene IDs with disease, for instance, associated with those genes, with the synonyms of those genes, with proteins, and with the metabolic pathways. And we perform a full index of Medline, and we provide an interface, well, similar to PubMed, not so efficient, unfortunately, but there is a different paradigm in here because we are using query expansion, and we are providing documents or publications that, for instance, may be publishing about the diseases that are associated to those genes. So it's a network of biological concepts. During these years, we have also been working on open source software. In here, we have been developing a platform that we call it Neji. So if someone of you wants to go deeper in text mining and make your own code, you don't need to start from scratch. There are several frameworks around here. One of those, very tuned for biomedical field, is our platform, Neji. So it's an open source that you can use, it processes the entire pipeline of text mining. And this means reading a document, splitting the document because the document can be in different files, XML file, a document, a PDF, different structures. So we strip all those formats and we convert it in pure text. Then we need to make sentence splitting, for instance, identifying the concepts, annotating the concepts. So there are several techniques that are available in this framework. Now you can see some final application of text mining. This is a public site that is available also in our webpage. And with BICAS, BICAS is just a concept recognition system that allows you to copy-paste a specific test that document that you have, and the system will annotate a set of concepts that you may select on the left side of this window. Systems not only annotate with different colors. For instance, I'm interested in diseases. Please annotate the name of diseases in this document, but also tells us what are the dictionary or the ontologies that were used to perform those annotations. You can also provide a PubMed ID, for instance, or search in PubMed, and system will do the retrieve of the medline for you and will annotate the abstract. You can use this application in 3 different ways. The first one is a web application as an end user, like in the previous slide. But if you construct software, you can also use a more programmatical interface that is a REST API for the ones that knows what it is, but basically you can build a program that dialogues with BICAS and that allows you programmatically to inquire our platform. And you can also integrate, even in your home page, a widget, because we also provide this as a widget that is more simple because it's just a code, a bunch of JavaScript that you can copy-paste in any web page. So the problem with the previous tool is that we are talking about automatic text mining identification and annotation. And the best tools, some of are from, we have also developed, we can achieve results between 80 and 90% of F measure that measures the quality of the tool. So this means that in the previous abstract, we can identify at least the maximum of 90% of concepts. So there is 10% or 20% that the computational approach is not enough. And this is the limit. So what is the solution for that? So we need curation, manual curation to go from that point. We developed, to answer this problem, we developed a collaborative curation tool that we call EGAS. This application allows to annotate the concepts, but also allows the user to annotate by themselves. I will show a brief demo of this tool. The main concept of this tool is the following. We have projects. Here you can see a set of projects. We create, if you want to annotate a set of, for instance, neurodegenerative diseases documents, we create a project for that. We can associate a set of rules that we are seeing now, a set of users also, who are the users that are available to see and to annotate the documents. A set of concepts, we have already a set of concepts. We can rename or group several concepts in new concept types. We can also modify the colors that we want to use in the annotation process. And besides the concept, this is novelty also of this solution, we are annotating relations, and we can choose the relations, for instance, a drag interaction. And this will be annotated with the tool. The second step is we have already in place the project, the users, the concepts, the events. Now we need to import the articles, and this can be manually, locally. I have already documents in my computer. Can be searching in PubMed, that is what is showing now. So I type a concept term, or I can use also the PubMed ID directly. So after I'm performing a search like in PubMed, the system will retrieve the ranked list of documents that are more associated with the concepts that I type, and then we select a set of documents and we can import. The system will import those documents into EGAS, and now you already saw the abstract, but it was divided in sentences, and now we select the corresponding concept tree that we want to use in the annotation. So this is just made once, and after that the system will annotate. This is already performed automatically. The system annotates the concepts. If the user, we are trying to curate the 10-20% that was not identified. In here, we identify a concept that was not there. So we mark this concept and we select the right ontology. Now we are using the event association. So we select two concepts and we create an event between those concepts. So another example that cross more than one line. So the first concept of the association and second concept, and what is the event or the section that I want to use. So this is a very interactive process, and at the end the system can export this in a well-known format that can be used outside EGAS. At any time we can select the concepts or the events that we want to see in the system if the window tends to be a bit confusing, for instance. We also have a collaborative, now we are scrolling across several documents because the system annotates all the documents automatically. The system has also a collaborative environment where, like a message or chat system, allows to interact with other curators that are at the same time annotating this document, and we can also see what the other curators are annotating, such in this case. So that was an highlight here that was not me as a user, but it was other colleague that annotates the system. With this tool, we participated last year in Washington, D.C. in BioCreative 4, that is the most well-known challenge for text mining. Each system was, we participate in a specific task that is called Use an Interactive Task. Each tool was evaluated by 4 curators. Each one used a total of 50 documents, half of them in a full automatical process and the other 25 with manual annotator. We are proud that EGAS ranked in the first place, considering the user experience and the final recommendation of the tool, quite easy to use. So, text mining challenge, what are our current challenge? I will not enumerate because we need to skip up and gain some time, but there are several challenges that we are trying to address right now, like relation mining, like AHR mining, and I will talk a bit more about in the second half, and social mining. For instance, one interesting study that we performed last year was to analyze how can we detect pandemics just processing Twitter message in Portugal and Spain. So it's quite a useful approach that shows how text mining computational algorithms can be applied to process more social media. So there are lots of people involved. Maybe I need to address particularly David Campos, that was one of the guys most active in the development of tools. So you may be wondering how text mining can help you in your daily work, because most of the time you don't have only scientific documents to read. Indeed, let's look at other kind of data. There is a fast-growing in healthcare data that you can realize in medical institutions are collecting patient data, electronic health data, cohorts, observational data. Pharmacology is also moving and trying to integrate more data in order to create more efficient medicines. Prescriptions are getting digital. So I will say that the entire landscape of healthcare is getting digital. And this is also creating a great opportunity to generate more data. So we are facilitating the data, but at the same time, we are creating more and more data. From the omics perspective, we also assist to the same problem. We have biobanks, we have genomics, transcriptomics, proteomics, metabolomics, next generation sequence databases. So there is a big data problem in this field. So we know that we have lots of data. Maybe this data has some value, but how can we exploit the value of this data? From an engineering perspective, or a computational science perspective, how can we dig into this data? So now I will describe a bit the effort that we have been conducting in a European project that is called EMIF. EMIF stands for European Medical Information Framework. It's a project founded by IMI. IMI is a joint undertaking initiative from the European Commission and the EFIA, that is the European Federation for Pharma Companies, Pharma Industry. This means that basically European Commission puts half of the budget in the overall project, and the other half is contribution from pharma companies in mankind. This is currently the bigger project in IMI, with almost 60 partners from all Europe, including universities, research organizations, public bodies, non-profit groups. What were the main motivations for this project? Imagine a world where researchers can pool available patient data level to gain new insights around disease etiologies and define new treatment targets, where we can optimize clinical R&D by, let's say, 50% using existing patient-level data, where new treatments are effective in more patients in 90–95%, where we can reduce treatments failures with timely prediction and timely intervention. We can continuously monitoring the risk-benefit profile of new therapies using naturalistic data, and it was already Professor Raquel already gave us an example of how the amount of subjects are quite important to statistically infer about the quality of a study. So the problem is that this means a lot of challenge. What are the challenges with the reuse of patient-level data? We have data gaps. So if we try to understand what is the available data in EHR systems, for instance, there are missing data elements. Sometimes we don't know the outcome of the patient. Some studies, some scientific research or research studies require detail that typically is not routinely connected. For instance, I need to have a body mass index for my patients during the last 10 years, and I realize that GPs are not collecting that data. Coding often occurs at fast level. Typically, if you use ICD-9, for instance, we miss granularity. 80% of information stored is in a structured way. Well, this is good for text mining. Data quality problem. We have a lack of longitudinal coherence. The way institutions collect data today is quite different from the way they collect 5 years ago or 10 years ago. Coding sometimes, well, most of the times occur for administrative reasons. So this means that there is a up-down coding. Most of the institutions code because the patients or the institutions needs to be reimbursed by the security system. So there is sometimes not a clear medical proposal on this coding. Coding occurs months after patient encounter. This means that if I want to use data today, I need to wait 6 months for coding. And coding is very important because coding is the way I can understand what disease a specific patient had. Why this happens? Because clinical institutions typically have specific service for coding. It's not the physician that performs the coding. And we need also to assure the data provenance because it's like a scientific paper. We know that the quality depends on the author. The third set of problems is related to semantics. What is this semantics issue? There are many standards, many versions. This is complex care. There are many healthcare professionals involved with many handovers. So this means basically that the notion of a treatment or the diagnostic of a disease, in some way it varies from country to country and from professional to professional. And finally, but not least, privacy, legal, security issues is quite a big problem. This is clear a top priority. There are different interpretations of what is patient-level data, what is the rights of the patient, what is the right of institution, what is the possible access for the researcher. So we need to build trust on those databases. So MF Vision tries to work around those set of problems. And the vision of the project, it's a project that started 1 year ago, 1 year and a half, is to be the trusted European hub to healthcare data intelligence, enable new insights into diseases and treatments. It's a complex organization. It's in fact a project that is composed by 3 different projects. I will call it 3 topics, not 3 projects. But the composition of the project indeed starts from 3 different calls that were combined into a single organization. The first one is MIF platform. The aim of this project is to build infrastructures that can help other researchers and other, I will say, disease-specific and oriented projects to rely upon. The second project is related to identifying biomarkers that allows to explain the metabolic complications in obesity. And the third one is related to Alzheimer's disease biomarkers. So some work that I will show you is mostly related to MIF platform. Also, as providers of the entire consortium, the tools that we are built will help metabolics and Alzheimer's disease projects to conduct their own research. Inside the consortium, we have already around 50 million patients that we are using as a proof of concept of our methodology. Those subjects come from 7 different countries, so there are lots of different ontology, different semantics that needs also to be addressed. MIF has several distinct data access levels. Basically, the problem is that I have a big database with 10 million patients. My business model during the last 10 years was made around this database. But on the other side of the room, there is a pharma company or another research group that wants to access my database. But we need to arrange a model that allows this sharing to be useful for both partners. So these three layers show our current approach. We are building what we call MIF catalog that allows to characterize this database in the best way as possible, that someone can Google in the catalog and understand that, okay, I will not have access to the persons, but I can understand the characteristics of this database. So if I want to conduct, let's say, Alzheimer or Parkinson's disease research, I know that database A, B, and C have patients that allows me to go further, deeper in this analysis. So I can arrange some commercial agreement, research agreement with them. So this strategy implies two different approaches, a twofold approach. One that is quite horizontal, is just a way to expose the general characteristics of the databases, and a second one that is more vertical, or is vertical, and means drill down at the patient level data. This means that we need to build secure environments where the research involved will not disclose the patient level data from outside this environment. In our group, we are building an important piece of MIF that is called the MIF catalog. And the MIF catalog is a way to describe what we call database fingerprints. Okay? We have basically input-output systems. The input can be manually on the left, can be with documents in any form, kind of data that we can process and index, and can be also in a programmatic way. And at the end, we have several ways to use the system. This is the current home page of the MIF catalog. Is available public under registration, but currently the registration is only is restricted to MIF partners. This is an example of database. We have already more than 50 databases in the system. The user can see what are the databases and can use what I call it fingerprint to characterize database. So let's say you have a database that you want to expose to the research community in order to, well, to better exploit your own database. So you can go inside MIF catalog and you can fulfill this questionnaire that is the fingerprinted database. And this questionnaire allows to, the aim of the questionnaire is to characterize the best as we can the database that you have, okay? At any time, besides the fingerprint, we have several tabs of information. This is the fingerprint structure, but we can also add population characteristics. It's a form of processing locally my database and extract aggregate data. And this is the second level of the access to the database. I can provide, for instance, how many patients I have with a specific disease that born during the last 2 years. This is the kind of thing that we are currently provide also. We have also a more technical API interface to the system. Basically, this means that automatically each database can update remotely the data because they are very dynamic also. This is an example that can be shown. Well, I will skip this one. We can provide, we can perform also queries like in Google, in any search engine, and the terms that you use for the query will be compared. We are indexing basically all the information that are collected, and we can retrieve the databases that, well, basically have something about to do about with the term that we use in the query. The previous one is a free text query, but we also provide more complex queries. For instance, suppose that you want to know what are the cohorts that are collecting CSF data and amyloid imaging at the same time. So this is type of query that typical research want to ask for a set of databases. And for that, we build a query builder. Query builder uses the structure of the fingerprint and allows the end user to create basically a Boolean expression graphically without typing anything. And at the end, it can save this and it can retrieve the databases. So after identifying the databases that answer their question, researchers can go deeper, a bit deeper, not too much, not too patient, into the information that is available. For instance, you can see the geolocation of the databases, some more administrative scientific information of each, if we cross inside the pinpoint. You can visualize aggregate data for this database, for instance, the number of TB patients during the last 20 years by gender, by age. So there are several fields that can be selected in here. This is another example, and there are many others. There is also the possibility to associate, and now we are showing the connectivity between text mining and this tool, to associate scientific publications related to this particular databases or set of problems. The user can compare the results, for instance, using the second column database as reference. He can see what are the differences for other databases and what are the commonalities, of course. Well, and during this last year and a half, we have been creating what we call EMIF community. So number of users have been growing, the number of databases, we are gathering information, for instance, what are the most popular search, what are the kind of use like Google Analytics that is performed in the system. Out of time, yeah? So there are also lots of credits to my dear. I will pass this one, I will pass this one, and thank you for your patience, and I will be happy to answer your questions.
So are there any questions for Professor? Okay, thank you very much, Professor.
Take this one.
So I'm going to introduce now our last speaker of morning session. So our last speaker are the Dr. Fernando Mota. The Dr. Mota is the global trial the leader at Novartis in Basel in Switzerland. He's the graduate of the Porto University, and after the graduation he also did the master's program there. Afterward, he had experience in the various positions in industry, and now he is also finishing his MBA along with the job in the Novartis, and he's going to speak about the very interesting topic about the overview on the developmental process of clinical trial. So I would like to welcome Dr. Fernando Mota.
So good morning and welcome to Porto. Thanks for the nice words and thanks for the organization for inviting me. But all that you have heard is that I'm the roadblock between you and lunch, so I will try to make this quick. I am a project manager. I work at Novartis. Thank you very much. So usually when people ask me what I do, I say that I work in clinical research. So I would ask you to raise your hand who thinks that I go into a basement, I put on a lab coat, and I use all these kinds of fancy recognition tools where I'm stuck and I cannot leave. So who thinks that clinical research is going to a basement? So if you can, come on, you can raise your hands. You still have an energy to do that. Okay, so it's not. Clinical research, it's much more than just what you think about working in a lab. I think, and one of the reasons I really was it was a big pleasure to participate here is that I want to make you all aware that in clinical trials you have a lot of different things you can do. I know that I don't have much time to go into a lot of depth, but we can talk tomorrow in the workshop more about what you can do in this area. So I'm going to try, because it's always a try, to give you an overview of what are exactly clinical trials, what is clinical research. So we're going a bit through some basic concepts. I will talk to you about something you probably heard at least during a 1-hour pharmacology lesson about the development phases. So I'm sure you've heard the concepts. I will talk to you about something that is the basis of clinical research and that we all should be aware. And I think it's a big gap that we have in most of the degrees in life sciences, especially the ones who can work in clinical trials, and that is good clinical practices. And this is not meaning that the doctors have to be in the hospital on time. It's much more than that. It's how to do clinical trials and what needs to be respected. Because one thing clinical trials are not, they are not crazy experiments by people sitting on basements and thinking and doing it without any kind of surveillance. So clinical trials are very well regulated. They have a very detailed set of rules, and it is very important for you to know that these rules exist. You will also see that during my presentation, one of the things that I will talk to you very lightly over the next 3 hours. Everyone is sleeping. No, it's only 40 minutes. So one of the things that I will talk to you about is legislation. And when you hear the word legislation, you all think that it's something that it's very dense, it's something that you don't want to get into. But that's the thing that will make the difference as professionals. You need to know the legislation, you need to know the rules, and that's why you will see that it's important that you try to get a bit more sensitive to know the legislation, because you will be professionals in your areas and you will need to be very comfortable with knowing what the legislation is and how to work with the legislation. So, going into what legislation is, does anyone know what a drug is, a medicine is? I mean, just you think any compound is a medicine? Do you think that something that does something to your body is a medicine? No, it's not. And because we don't always know exactly how and how to define things, we have to go back to legislation. And we do have a directive that clearly states what is the definition of a medicinal product. You as pharmacists should know this by heart, because it's your bread and water. So don't hold back on looking at legislation, on knowing the concepts, because you will be judged as professionals to know exactly what the concepts are. So homework, do go back, look where the legislation is, and I hope that the ones of you coming to the workshop will know this tomorrow. Why do we do clinical research? So first of all, it's pharmaceutical companies' business. Pharmaceutical companies develop drugs because they later will sell the drugs and they will make money out of it. And there is no shame in that. On top of that, there are also other reasons why drugs are developed. Nowadays, and more and more, we have orphan drugs, drugs that don't necessarily generate enough revenue for paying for their development. These drugs are also necessary, and we need to work and we need to find ways on how to develop drugs that will not be profitable. So we develop drugs if we find a treatment need, if we can see that the drug will have some benefit. Would that benefit be health? Yes. Sometimes we also need to know that our drugs will be sold to hospitals, to governments, to countries. If we can prove that our drugs will somehow have also other benefits, like reduce the health costs. How much does a country earn if they have their full working force working every day rather than being affected by a certain kind of disease? So you've heard all about data mining, you've heard all about looking at the data, and nowadays it's even more important that we prove that our drugs bring some kind of benefits. Financial for the governments, benefit for the health of the patients. All this will come into place when we think about how to develop drugs. I have a very loud voice, so I'm not sure if you hear me better with the mic or without the mic. Okay, with the mic. Okay. Over the last thousands of years, we survived without medicines. We would die at the age of 35, but we didn't have medicines by the beginning of when 300, 400 years, the medicines were the medicine knowledge was very little. Given in the last, I would say, 100 years, we've almost doubled our life expectancy. A big part of that has to do with the development of not only medicines, but all the availability of healthcare therapies. When I look at 10 years ago when I finished university, we didn't even have Ryanair. So if I wanted to go anywhere, it was very difficult, very expensive. That's why this is my first IPSF. I always wanted to go, but it was not always easy to go everywhere. Same thing happens with medicines. Lately, the development, the tools available, the fact that we have more ability of processing data has made the world in clinical trials more challenging, but also more effective. As you don't remember, it's not of your days, but we did have a time where there was no approval of medicines. You could just find something that would work, sell it in your local market, and you were an apothecarian, and that would solve a lot of the local healthcare problems. Nowadays, it's no longer like that. Developing drugs, it's a very lengthy, it's a very intensive, it's a heavily regulated process, and it involves a lot of professionals so that we can actually reach a drug to market. Drugs don't always come out of the bench. They are not all synthesized. Some of them are coming from plants. Some of them are actually inorganic. But it is a fact that more and more we're looking at industrial processes to produce drugs. And more we are going into the biotechnological arena, where it's becoming, not only it requires more people, it requires more skilled people, and also because most of our basic needs, our big and widespread diseases are already covered by the existing drugs. So we need to go into a healthcare world where personalized medicine will take place. So the drugs will no longer be developed for the whole population, but you will get, I don't think it's going to be in our time, but a drug will be made just for you, for your disease. That's really the aim of developing drugs, and I think it will be a possibility. Maybe I will not see it, but I believe it will be a possibility. So research doesn't only happen at universities. Research also happens with pharma companies. Governmental institutions also do clinical research. So it is important for you to know that there is a very wide area where clinical research is happening. Now, numbers, money. How much does it cost to develop a drug? No one really knows how much it really costs to develop a drug. It is estimate, and the last estimate says that it can cost up to $2.1 billion to develop a single drug. Now, this is always a matter of viewpoint, because only big pharma companies can develop drugs that cost $2.1 billion. Smaller companies can develop drugs with as much as $300,000, $400,000. I've been working for a Portuguese pharmaceutical company where we developed a drug here in Portugal, and it was a very big effort. Overall costs, maybe almost €500,000, but it was possible. So read behind the lines. The average cost could reach up to $2.1 billion, and that's a fact. There are drugs for whom the cost was as high as 1, 2, 3, 4, 5 billions, and the revenue that the companies had with that was zero. Because you will see that it's a very risky process. Out of thousands of tested compounds, only one will come through as a winner. I know that you all hear about Google, Apple, Steve Jobs, and how much all these fancy companies spend in research. Nothing compares to pharmaceutical R&D. This is the percentage of the money that the companies make that they reinvest in research. If you think of very fancy planes that have pools in the first class and all that is involved around that, the airplane industry spends 1.2% in research. Pharmaceutical companies spend about 17%, and this is an average. There are companies spending over 20%, and this is a huge investment. This is a huge risk for a company, because remember, there is a very high likelihood that many of them will fail. So you will see that also one of the focus of clinical research is to kill the drug as soon as possible. We will do everything to make sure that if there is the slightest hint that it will not work, we will not throw any more money into it. This is a funnel, and this is exactly, I will show you a couple of pictures. They are maybe 10 years different, but it's always the same. It's a very tight funnel. You will see in the summary, but just to give you a glimpse, the average is that you synthesize over 10,000 compounds to get one approved drug. So the clinical trial development, it's not a linear process. You have heard about probably phase 0, phase 1, or let's start from the beginning. You've heard about in vitro, in vivo, animal, preclinical, phase 1, phase 2. That's not the case. This slide, I have it for several years, and it's just to show you how many things are happening in parallel and how much all of you can contribute depending on what you like to do. When a drug is being developed, not only clinical trials are happening. We want to deliver the drugs in the way that it's more suitable for your patients. We want to deliver the drugs in a way that it's more easy for the patients to remember how to take those drugs. We want the drugs to have a longer expiry period. We want them to be cheaper to produce. To give you a hint, there are several drugs that were perfect candidates with a huge potential that didn't go through just because they were too expensive to make. And the price to put them into the market was so high that no one would buy them. So they were good drugs, but they were not drugs that could reach the market. There was no benefit in doing that investment. So this is always a game that we have to play, because at the end, we have to minimize the risk we have when we bring a drug to market. The people who stay in the labs, that's not my case, but some people do stay in the labs. Some people are always trying to find ways to get new compounds. There are thousands of techniques. There are millions of ways to do so. Here are some hints of some things that you can do to get drugs. Some of the experts you will meet and hear during the next days will give you hundreds of other techniques. So there is always things happening, new ways to bring new drugs and better drugs. So don't get stuck to textbook numbering of ways to do drugs, but this is just a way of doing. These are just a few examples. More and more computers, bioinformatics are doing what we are now called in silico testing. When I was developing one drug for pain, going from one phase to the other, we saw a big pharma company who skipped the phase. We could not see any literature on how they skipped that phase. They actually did about 500 clinical trials, and that's what they said in one paper. They did 500 clinical trials. Now, if each clinical trial would cost at least $15 to $20 million, that was a huge amount of money. We dig deeper and we found that because they had the first generation and the second generation of that drug, they actually simulated clinical trials. And they could do, with very potent computers and servers, about 500 clinical trials in silico. So the technology exists. There are already companies doing simulations using computers, using clinical trials. I don't think it will replace in the nearby times doing actual clinical research, but at least they could get some idea of how to go to the next phase. Okay, where does it start? So we talked about synthesizing. So the first step, and do remember that the higher you go in the development, the more expensive it turns. So the first step usually is to do the testing in vitro. So we try to evaluate through several models if the compound is really effective, and more important, if the compound is safe. Then we go to in vivo. More and more we try to minimize the number of in vivo testing we do, but still this is an important step we have to take. We would not risk going with a drug to human clinical trials without having some basic pharmacological or toxicological idea. So this is why sometimes we actually have to do these tests. And again, to give you a picture of how much it costs just to start to develop a drug. So if we think we have a compound that has some potential, we would do some basic in vivo testing. These are basic testings. We're not talking about anything very much advanced or very complex. If you sum up how much it costs, the basic development, and this is a maybe 2-year-old costs, $5 million are spent just knowing if a drug is good enough to go into human clinical trials. When we are doing the animal testing, it is also very important for us to know how the drug behaves. You will hear this term, and please note that my objective is not to be extensive, detailed about everything you need to know about clinical trials, but some important concepts. And you'll hear this, and you probably have heard this, ADME, and this is very important. This gives us a hint of if the drug will actually be usable. There were drugs which were very promising candidates, which when given for the first time in humans, they completely vanished. They entered the human body and there was no effect. And this is what we try to minimize by studying the drug before doing those first trials. One thing that is very important and that we all need to know is that there is a lot of confidentiality around. So the learning from one drug to the other doesn't really exist within companies, within institutions. More and more there are rules that make you publish your data, and this is why the previous presentation shows something that is very interesting, is if we actually share databases, we can actually save a lot of money in future development by knowing what other people did. This doesn't really happen so much within the pharmaceutical industry. Now, clinical research, and this is really where pharmacists come into place, formulation design. This is also a part of clinical research. When you are given a drug to develop, sometimes you have to give it in a capsule that it's bigger than your throat. There is really no way you can bring a drug to market in that kind of conditions. Most of our colleagues spend their life trying to find ways on how to make your medicines more easy to take, more possible to take, how to make your patients more compliant on how to take drugs. I remember about 7 years ago, one of the big steps moving forward was that we actually could print a moon and a sun on the blisters so that we could remember the patients what was the drug to be taken in the morning and in the evening. So these are evolution, this is research that is constantly happening and where you can also take a part. Now, your drug went through all the phases, you actually have something you can give to humans, right? So what do? Your drug is effective, at least it looks like from the animal models you tested it on. Doesn't have any hints of being non-safe, because that's what it is at the moment. You don't see hints of being non-safe, you don't think it's safe yet. So you go for it. You go for your first for your first trials, for your first in humans. The time that passed since you first had the compound in your hands is something from 3 to 5 years. So now, after 3 to 5 years, you are able to say, okay, I have something that I can test on humans. One of about every 10,000, it's a zero missing, one out of every 10,000 compounds does actually go into human trials, but only one. So again, legislation. I'm not expecting you to read all this. I'm not expecting you to fall asleep reading all this, because I all know that you need a good injection of caffeine. But do know that it is important, and that will be the basis of most of your work. Don't look at sideways. It's important. There is a regulation that actually defines what a clinical trial is. There are people who actually meet and come up with these nice sentences. And they have some meaning for some reasons. So do explore this when you enter any area of expertise, because legislation, and it's really the basis of your work. You will give you the hint on what you can and cannot do. So this is the traditional phases of clinical trials. Nowadays we have phase 0, we have a lot of other variations of clinical trial development. It is not my intent to go in that detail. From what I know, from when I did pharmacy, from what I know from the colleagues, your colleagues I spoke to today, there are not so many basics around clinical trials. You do have a big puzzle, you have a lot of pieces, but they were not put together yet. I don't want to go in too much detail, but these are the traditional phases. So from phase 1, 2, 3, and 4. So what do we do in the phase 1? So one question, so that we try to... So has everyone here heard about... So let's make the question differently. Please raise your hand if you ever heard about clinical trials and clinical phases. Okay, please raise your hand if you haven't heard. Ah, that's good. So, okay, so I can go now and we can all go to lunch, okay? Okay, phase 1, it's really the first phase. Phase 1 trials don't only happen in the beginning. Phase 1 trials keep occurring as the drug is developed. Because the difference from a phase 1 to the phase 2 and 3 trials is that phase 1 is done on healthy volunteers. The reason why healthy volunteers are used is because we can really get a more precise picture of what happens with the drug. So if you want to do drug-drug interaction studies, so phase 1, it's not only the first trial that you do to start with a drug into humans. Phase 1 is everything where not patients are used, but actual healthy volunteers are used. They usually use a small amount of patients. They are done in a control setting, so usually in specialized units. And it's really to know about what your body does to the drug. Okay, so what's the bioavailability, some idea of pharmacodynamics, some idea of pharmacokinetics. You try to get the hints of the maximum dose that a patient can take. So this usually adds to the previous 3 to 5 years, another 2 years, so that you can have some data to know, should we go to phase 2? As I said, our target is to kill the drug if we see any hint that it will not work as soon as possible. So what do we do in phase 2? And phase 2 is now more and more divided in 2A and 2B. Basically, it means how big the phase 2 is. So here we are starting with giving our drugs to patients. And the main target of the phase 2 is actually to define a dosage, to know in the later trials where more people are needed, how much should we give to each patient. So a phase 2 is a proof of concept. We try to understand what would be the dosages we should use. We try to answer some basic questions. And most of all, we try to understand what actually happens to the drug in actual patients. So we can also see if using it in patients rather than in healthy volunteers, if there are any changes to their pharmacokinetics. And we also collect a lot of safety data, because that's really important for us. One of the things that will immediately take your development out of the picture is if a hint of a drug not being safe comes up. No one will want to invest in a drug that it's not safe, which is a good thing, because then you don't get, or you do have a good filter for getting unsafe drugs out to the market. So when you went through all this difficult process, 7, 8, 10 years have passed, and now you have an idea of how your drug works. You have a picture of what would be an effective dose, and this is when you go to the phase 3. So up to phase 2, it's all about the sponsor, the person investing or the company investing. When you get to phase 3, this is your roadblock to get the drug approved to market. This is the time when you start talking with the authorities. It's not what you think the drug should do, it's not how you think you should develop, it's how you're going to develop a drug that your authorities, that the community will accept to be a drug to be approved. At this stage, we are ramping up the costs. We're going from 100 patients to 1,000, to 10,000 patients. We are escalating from $10 million to 100, to $800 million investing in development. So this is the time where to sign off on the go or no go, that's when we really need to take, a leap of faith. Developing drugs, it's sometimes also a gut feeling. Some people do push the drugs through because they believe in them, and they most important, they persuade investors, they persuade managers to go ahead with the drugs. So there is a bit of a leap of faith all the time. But it's a leap of faith with a lot of documentation in the back, okay? So it's not like playing the lotto. So these studies are longer in duration. These studies, they try to be, or they are certainly more robust. They will have to prove the effectiveness of the drug, and this is why you will see really an escalation in the number of patients. Because as you have heard, and you will always hear, it is important that your data is robust and that your data is believable. And the worst thing, and believe me, I've been in that situation, is when you get to the end of investing $30–40 million and you don't know if the drug works or not. And that happens. You don't get an answer to your question. You don't know if, is it really working? This is what you really don't want to come down to. And it's very difficult when you look at your planning and you think, well, we might have had another 100 patients and probably we would get an answer. So this is where, some other people are working, statistics. And this is really when that small line that you see in the protocols comes and makes all the difference. So it happens. Sometimes you get to the end of a clinical trial, you see the drug is safe, but you don't know. You cannot say it didn't work. If you put the data like this, it seems that it works. If you put it like this, you don't really believe it works. So this is the worst thing that can happen, and this is why so much planning is involved around developing trials. When you reach to the end of phase 3, this is not the first time you talk with the authorities, but this is when you pull all your data and you go to EMEA, to FDA, to whatever agency and say, well, we suppose that we already had some conversation with them. We're giving you what you want. Do you give us the stamp to actually bring the money back that we invested or not? This is not an easy process. It's not an instant process. It can take up to 3, 4, 5 years. The companies will have further questions. Sometimes the drugs are approved, but still more investigation is required. Or let's say it, always more investigation will happen even after the drug is approved, but sometimes you are obliged by the authorities to do so. So it's not the end of development when you finish your phase 3. However, when your drug is approved, you come to the more pharmacoeconomic part of the studies. Phase 4 studies are studies that are not so robust. They are studies that are more open. They usually are more focused on medical needs, on trying to answer questions to some of them are just based on a country feeling or answering to a question specific to a country. So they are more they are not so much in the development umbrella, but more in the marketing umbrella. So how can we differentiate our drug compared to other drugs that are in the market? So there is a big focus on how to make your drug better than your competitors. And phase 4, it's much more focused on that, on trying to understand how you can make your drug shine when compared to their competitors. They're also very much related to safety. These studies are more light in what regards the intensity of the trials, but they're also very important because they do bring a lot of questions, they do bring a lot of answers that were not answered during the phase 3 trials. Don't forget that during a phase 3 trial, you're also limited. You cannot make it too complex. Don't forget that you have to go, and you should go, to real doctors that on top of doing their own research, doing their own practice, they also are doing research as a plus. So you cannot make your protocols so complex, so time intensive, so you cannot put everything into your phase 3. So that's when you bring some of your scales, some of your quality of life endpoints to the phase 4. Nowadays, the pharmaceutical industry is facing a big challenge all over, and as you study or hear more about drug development, you will see that companies are facing a lot of difficulties to actually bring drugs to the market. So this is also, and I'm here speaking as Fernando Mota, not as an employee of Novartis, but big pharma companies also tended to make the drug process more complex, more difficult, because smaller companies would not be so easily going into that game. That made it very heavy, that made it very difficult, and now there is a very high price to pay. Seeing that, the authorities have created, in the FDA it's called the Critical Path Initiative. Basically it's an accelerated development. If you actually come with a drug that in the very early phases you prove to be very effective and a very big need for the patient population, you are actually given the approval to go ahead and start marketing the drug. Of course, this is not a stamp and you can just do whatever you want. It's very heavily regulated. There is very strict, it's very strict what you can do with that drug, but still some patients get the drug sometimes 5, 6, 7 years earlier than what it would normally happen. So as a small summary, I think that this sheds a bit of light in the research and development process. So when you start really in the bench, you can screen up to 10,000 compounds. Out of those 10,000 compounds, roughly 250 will actually reach preclinical. Out of those 250 that reach preclinical, only 5 will actually go through phase 1. So there is a lot of waste. It's not a very ecological process, but this is how you have to do it. And especially many drugs fail, not only because they are not effective. So many drugs, that's what brings them further. They do show effectiveness, but what really brings drugs down is the safety part. So this is just, some interesting authorities and sites. They are interesting because their sites have a lot of information, not only about drug development. I would not recommend the Japanese, but not because it's not good, it's just because you cannot read it. But these are the 3 main authorities in the world, the 3 main regions. The reason why I'm talking about these 3 authorities, it's because it will come here, what is GCP? GCP, or Good Clinical Practices, came out as a need to standardize the drug development across the world. If you develop a drug in Europe, you want it to be approved in Japan, in the US, mostly because these are the major markets in the world. Now, some years ago, the standards were completely different from region to region. It doesn't mean that now they are not different. It doesn't mean that now we don't have to have a specific development process or program for each of these authorities. There are some very different approaches in these regions. But for the most part, the GCP is followed. And for the most part, at least the basic operational aspects of conducting clinical trials are similar. They will be different in what they expect to see as an endpoint, as a result, as how they see it's effective or not. But in how to conduct clinical trials, the systems, the way they are seen, are very much standardized across the whole world. GCP have 2 major pillars, and you'll hear a lot about clinical practice. One of them is the quality of the data, and the other is the ethical conduct. Clinical trials are very regulated. Before you get a clinical trial approved, you will at least have to go through at least two different approaches. One is the health authority. They will go to see if you have all the papers in place. They will look at endpoints if what you're studying makes sense. But most importantly, it will go through an ethics committee, and they will not approve the drug if it's not. They will not approve the trial if it's not scientifically sound. So you cannot do a study just because you want to do it. There has to be a rationale for it. At least that's why the ethics are there. So clinical trials not only have to answer the scientific needs, but they also have to be ethical. You cannot make your patients have a huge burden. You cannot take them blood every single minute just because it looks better on the chart. So this is where ethics come into place, and this is where it's really important that you have an idea that clinical trial, it's not just someone who thought of doing something. No, they do have to go through a lot of scrutiny before getting approved. Okay, so this was just a very quick overview. I know that you are all very hungry, so before you start throwing things at me, I will give you just 1 or 2 minutes for questions, or should we just wrap up and go for lunch? Thank you.
Thanks.
Okay. One question from Wales? No.
Close. Michael McIntyre, Naps, Australia. I was going to say. Yeah, I don't think there's really anybody in the room that will disagree that the clinical trial process is important for establishing safety and efficacy of new drugs. But do you think in any way that the process sort of stands in the way of the innovation process?
I think that the clinical trial process will always be under review. I think that there is a lot of openness from the companies to try to make it, or try not to be in the way of innovation. The smaller companies, the ones that are more open to take the risks, they cannot really invent the wheel because they don't really have the power to go and to an authority with something and not get approved. So there is a lot of money involved. When you have your drug and you want it approved, your main barrier is to go through the authorities. So within the authorities, there is a lot of openness to discuss and to make it more, or to be more innovative and not be in the way of innovation. However, they also have an important filter. Don't forget that thalidomide was not approved in the US because they stepped the feet down and said, thalidomide doesn't come here. So authorities are here in a very difficult balance. I do believe that sometimes there are a lot of things in the way of innovation. Of course, that the authorities have an important responsibility, but I don't think that's really the major step in the way of innovation.
Thank you for the time.
Okay, so thank you very much and have a good meeting.
Thank you very much. So it's going to be the end of the morning session. I think now you are all hungry, so please proceed to the lunch. At the timings? Okay, so 1 minute, there is an announcement.
So guys, I just have some announcements. I hope that you can hear me. So for tonight, we have the regional assemblies, so don't forget it, okay? And there's no dinner at the canteen, so it's going to be the free night. You can just explore Porto and some bars or just relax a little bit. Regarding to regional assemblies, it's compulsory for the official delegates, don't forget it, okay? So, and then it starts at 8 p.m., okay? For the ODs from Paro Assembly, please find Lucas, the chairperson of Paro, okay, to speak with him. And for the international night and auction night, if you have some items, you should deliver it to Sheena, okay? The exec, you know Sheena, okay. And if you want to hear some songs as well in these 2 nights, you should email her, okay, with the songs. Regarding the poster exhibition competition, that it's going to take place at 2 p.m. until 3 p.m. So people that have to do the presentation have to be here at 2 p.m. And at the canteen you have 2 tables reserved to be more quick, okay? So good lunch and see you at 3 p.m. here to start the afternoon session. Thank you. Good afternoon, guys. It's too loud. I'm going to ask you to sit, please, to begin with the afternoon session. Please hurry a little bit. Come, come, come. Hurry up. Hope that you enjoy your lunch. So we are going to start the afternoon session for the Scientific Symposium. I would like to introduce to you Dr. Emma Paulino. She's going to be the chair of this afternoon session. Dr. Emma Paulino, she's a Portuguese graduated pharmacist and professional secretary of member of the Executive Committee for the International Pharmaceutical Federation. She is the actual director of the Projects and Services Department in Holland's Pharmacies and president of the Lisbon and South Branch in Portuguese Pharmaceutical Society as well. She was a researcher in international collaboration on facilitators of practice change in community pharmacy from 2003 to 2009. Please welcome Dr. Emma Paulino.
Good afternoon. I hope you had a nice lunch after a very interesting morning session, and now we are going to continue our session with a new panel where I will introduce the next speaker, and afterwards we will be able to have a roundtable discussion with the presenters from this morning, where you will be able to ask some questions and maybe participate in a little bit of debate on where the advancements in the translational science will lead us in the future. But I will proceed with introducing the next speaker for the afternoon. It's Dr. André Albergaria. He's the director of the Patimup Translational Research Unit. In terms of education, André obtained his master's degree in molecular oncology from the Faculty of Medicine of Porto University, and his PhD was developed in joining collaboration between the Institute of Life Sciences and Health from the University of Minho, Patimup, and Imperial College School of Medicine in London, England, where he was a PhD resident student between 2006 and 2008. André Albergaria is a specialist in molecular oncology and a postdoctoral fellow at Patimup. His scientific expertise is focused on breast cancer, mainly on genetic and epigenetic regulatory mechanisms, endocrine resistance, and on the identification of new prognostic tools for managing breast cancer. He is now the director of the Patimup Translational Research Unit, and he will present the work that has been done at this unit with the title The New Era of Translational Science, Recent Advances in Therapeutics and Biomarkers. Thank you very much, and please welcome Andrea Albergaria to the stage. Thank you.
So before my images starts doing the panel, yeah. So good afternoon for everyone. Thank you for the introduction, and especially thank you for this invitation. I was invited to give a talk on this theme of translation research and personalized medicine. Actually, I immediately accepted this invitation since this is a subject that I'm involved in the last years. Actually, the term translation research, personalized medicine, precision medicine, as some British used to call it, is in the mouth of most of the researchers that are doing their job more related with clinical aspects. The basic researchers don't, but those researchers that are more related with disease research, nowadays talking about translation research and personalized medicine very often. However, this is a topic not so easy to define, and I think that today I'm sure that you already have very good insights about what is translation research and why translation research is important. As Emma said, I'm a researcher, so I'll give you some insights about how translation research is seen by academia point of view, but also how this could be integrated in pharma and clinicians, and how this interplay is so important for the next few years we will going to live. So the title of my presentation is The New Era of Translation Research, and then I will have the opportunity to discuss with you some and show you some recent advances in therapeutics and biomarkers that came in the last few years. So why everyone is talking about translation research and personalized medicine? Because of this simple fact. There's a lot of reasons for translation research and personalized medicine, but basically the problem is that the current developments in basic discovery sciences have not been mirrored by the same level of progression in understanding the clinical basis of disease, and therefore we don't have enough development of novel, effective therapies. And the word effective here is really important, as you can see further on in this presentation. So all the players on this landscape are arguing with each other, and there are three main players on this whole system of translation research. For one side, of course, you have always the patient in the center of all the system as the most important thing that we should care about. But one side you have research institutions that always arguing why clinicians not talking with us more, why industry is just playing their game inside their headquarters and not are so close to academia. But industry, healthcare industry will say, well, clinicians are not doing enough clinical research, so we don't know which are the problems inside the clinic so we can address them as well as we can. And the clinicians say, we are not studying. The guys who should study is academia, so the guys not coming to us and trying to solve our problems. The good thing in all of this is that these three parts are talking more and more and more to create what I call a patient-centered, knowledge-driven ecosystem that aims to generate, contribute, manage, and analyze data available from all parts of this landscape to accelerate proof of concept. So that's what we want, is to have these three parts of the landscape talking together, trying to solve and try to inject more innovation in the health system. It is a shift of a paradigm, yes, it is. Basically, translation research is not more than transduce the knowledge that is created and generated in basic science, pharma, information technology, engineer the proper medicine into human health through the market that is also related with pharma companies. So for me, this is the most robust definition of translation research or translation medicine, as you prefer. So basically, it seems that is an answer for a simple problem. Basically, what we want to do is a bridge between basic science and clinical research, basically to take our knowledge from the bench and deliver it to our patients. I would say that some institutions around the world are trying to do this move with a little bit less success, and some of them are actually failing this because they forgot some details and some support stones that are needed to implement personalized medicine, as we will discuss further on. So I would say that is actually an answer for a not simple problem. You can see translation research in different perspectives. You can see from the clinical perspective, you can see from the research institute that includes academia, and we also can see it from the healthcare industry. You can see it in different approaches. You can make your own path to reach the inner of the labyrinth, but there's a common reason for everything and for all these perspectives. Money is the center of this issue for a positive way. This is not bad, okay? This money, but it's no shame in to say it. Money is the center. If you talk with academia, if you talk with patients and oncologists, if you talk industry, money is a transversal common reason for impelling translation research forward. And I will explain you why. Why? Because the market is actually driving this revolution towards translation research. Because healthcare and health research is the economic sector that, I would say, burn more investment, as you know. there's a lot of investments in R&D in biotech and pharmaceutical companies. In 2011, half of the leading global R&D companies were pharmaceutical companies. So we are spending a lot of money doing research in the benefit of our patients. So we are creating this very heavy health economic environment. So let's see some facts from the industry. For one side, as you know, maybe better than me because you are students from pharma, the majority of tested compounds do not progress from drug developers' laboratories to patient bedsides. As already was shown today by Fernando, if you have 5,000 to 10,000 early phase research compounds, after 12 or 15 or 16 years, you'd end up with one compound into the market. So it takes a long time with a lot of money. And the tested products entering in phase 1 trial and achieving FDA approval market is actually still decreasing in the last years. So there's the number of new chemical or biological entities are coming down in the last year. So we have fewer drugs and fewer biologicals injected into the pharma market. This is a much simpler graphic, so you can see there's a huge increase in R&D spend, with new molecular entities coming down year after year until this is a graphic from 2009, but here are 2012, so it's more complete here, but more simple to analyze in the other. And on top of this, we know that the estimated full cost of bringing a new chemical or biological entity to the market is coming up every single year. So there's an increased development costs also due to the regulatory issues, because regulatory agencies are more and more restrict in the clinical trials so they can approve a drug to the market. So it costs an average of about $1.5 billion to develop a single drug. So the costs are really, really high. But at the end of the day, we have this, I would say, a paradox. In a world where failure is many times more likely than success, that is in biotech and pharma, and where there's a huge decrease of incoming funds, also by the coming of generic emerging markets that are building drugs in a much cheaper cost. That's where pharmaceutical companies start to planning their success, doing a lot of development and exploratory projects, with many designed to jumpstart and boost their innovation objectives. This is where the objective execute programs in an accurate and cost-effective decision-making with respect to target mechanism. That means, in other words, they want projects to make them de-risk their projects before they start or when they start the clinical trials. So they need to do exploratory projects with academia before they go to clinical trials, because when they go to clinical trials, they are reducing the risk if they already know some information about biomarkers and disease behavior. So for me, that's absolutely the definition or the concept of translation research for the side of pharma industry. Actually, in fact, translation research and the new era of translation research is very appealing for pharma companies because it can enhance many aspects of pharmaceutical business. On one side, it increases the confidence in the rationale, because that's the rationale that they need before they start investing a huge amount of money on the clinical trials. And it will support the mechanism approach at much earlier stage than before. It improves drug candidates' survival and overall productivity of the drug development process. So basically what this means is that they can reuse and continuously investigate in some compounds that are on the shelves, giving these compounds to outsource research in very, very good reference centers and research centers to dig deeper on the utility of those compounds to be used in other diseases. So this is some of the comments from people that are quite involved in big pharma. Big pharma begins outsourcing research and development. This is a big shift in pharma reality compared in some years before. So here, as you can see, pharma, with less incumbent funds to spend on finding new deposits, they start thinking creatively. And actually the term outsourcing now should include all the partnerships, collaborations, alliances, and any other type similar relationships that combined will make this very productive network partnership. The other reason, it's obvious. Actually, this publication was in June 2011, and if nothing was done in some of these companies with new innovation, some of them will be in trouble at this moment. So the pharmaceutical industry is actually seeking stronger ties with academia because they are now experiencing a slew of patent expires, revenues. Therefore, the revenues will come much more exposed to competition from generic drugs. So they urgently need to inject more innovation into their pipelines, and they are taking this innovation or seeking this innovation where the innovation exists, that is academia and research institutions. So now don't forget the first image. The patient is our unifying agent, is our unifying issue. So let's see that one side you have the research-based pharmaceutical industry currently spending over 140 or almost $140 billion in R&D per year. But the truth is that from the side of the patient and the side of the oncology, we are still facing ineffective treatments, especially in oncology. I'm talking about oncology specifically, that's my area. So from the side of the patient and the side of the clinicians, the problem remains. We have very ineffective treatments, although all the progress we did in the last 30 or 40 years, that's definitely true. And among the patients that respond to therapies, some of them have severe side effects that also should be treated. But these side effects are related with decreased quality of life of our patients. Because the problem here is that this leads with a high cost of cancer care in the world. The costs of cancer care are huge, are among $900 billion per year only in the United States. And I can assure you that a third of this money, let's say $300 billion, are spent into treat severe side effects in patients that do not respond to oncology drugs. So at the top of the problem that they're associated with the problem, they are not responding to the treatment, the government is even paying to treat their side effects. So this should stop, and we should start doing better drugs to this patient and say, no, you don't go to the first-line therapy because you have this profile, then you go to the third-line therapy and avoid your side effects. So basically, we need better tailored treatments. Better tailored treatments that is responsible of everyone in this landscape, but also will be developed by pharmaceutical companies. So we have this cycling that we need to stop and take some actions that has been done across the world with translation and personalized medicine. So the term tailored medicine takes us to the ending point of translation research, that is personalized medicine. It's the most important. And personalized medicine means knowing what works, knowing why it works, knowing who it works for, and applying this knowledge to the center of our problem, that is the patients. So personalized medicine is basically understand of how a person's unique molecular and genetic profile makes them susceptible to certain disease, and also to predict which medical treatments will be safe and the ones will not be. This is really important. Personalized medicine is something that an institute decided to do or an hospital decided to do. This is a political decision. This involves a lot of millions that should be implemented in the country to do that. So there's a lot of stakeholders that should be called to this discussion, starting from the patients, the clinicians, pharma sector, faculty, universities, the investors, payers and providers. Remember, the insurance companies has a word to say there on here. Regulatory agencies, all of them should play an important role. And we should not forget one of those entities that we love every day and the ones that take real decisions and to decide to implement or not new systems of health. That is our beloved clinicians, politicians. Okay, so let's talk about some cornerstones of personalized medicine. Of course, that there's a lot of other important issues that should be taken into account to implement personalized medicine, but I just select some of the ones that I think that are really important. First of all, biomarkers, investment in biomarkers. Secondly, omics platforms. Third, world-class basic science. I won't talk about this because we don't have time. Everyone knows what means world-class basic science. Biobanks, and in the end, but not less important, information technology, bioinformatics. Information technology develops systems that can join all the information of each patient in the in unique databases that anyone, the clinicians and the researchers can have access to better design their experiments and to better design their clinical trials. Is absolutely important. I won't focus too much in this last compound. All these structures should be connected or linked by interface structures inside academia, inside the pharma, and inside the hospitals. Biomarkers. Let's talk about biomarkers first. Why biomarkers is so important? Because you actually need to better identify the patients who need each therapy. As you can see here, cancer is one, or at least the disease with the drug response rate lower, with lowest response drug rate. Although we were doing a lot of progress on that. So you basically don't need to stratify too much when you want to give analgesics to someone to have pain. If you have pain, you take this or that analgesics and you will be okay. It can take 2 hours, 3 hours, 5 hours, could be more or less effective, but any of analgesics will be okay. So you don't need stratified patients that will respond better or not. In cancer, it's absolutely the other way around. Cancer, we are different from each other, so our cancers are also different from each other. So it's one size does not fit all. You need better drugs to fit in each type, in each subset of patients. So we need to fill this gap and increase the drug response rate in cancer by doing stratification. And that's why biomarkers are so important. So from the side of oncology, personalized medicine can be resumed on these 3-4 steps. First, you genotype. Then you select the target. Then you monitor early response. So when you have a patient, you should go inside the patient, take the biopsy, take the tumor, analyze it by histopathology, by genotyping, by analyzing the pathways that are active or not. Then you select the perfect and not perfect, the better treatment for this patient, and then you go to early monitorization of the patient. So the biomarkers came to be the end of one size fits all. Let's see what we were in before. You have a population on the left-hand side, the first image. So you have a population, let's say, with gastric cancer. And you have 1 or 2 suitable drugs for gastric cancer. And you treat those population of people, of patients, with these drugs. In the end of 6 months, 1 year, 2 years, you actually have a subset of patients that actually are quite okay, they are very happy with their families, and they have their disease well controlled, or even they are cured. So they are the good responders. They respond to the treatment. But you still have a significant amount of patients that are the non-responders. Most of these non-responders will die during this process. We won't have their disease controlled, and at the top of that, we'll have adverse drug events during this time. Biomarkers just had a step before the decision of which therapeutics this patient should have. So biomarkers will help you to choose the right drug in the right dose for the right patient. So you are avoiding to treat patients with severe side effects. You are saving time in patients that sometimes the time is not too big, and you are shortening the time of treatment. This is really important. So the biomarker-based prediction of treatment response is an approach that provides the ability to classify individuals in subpopulations that differ in their response to specific treatment. Non-responders, as I said, are really important in oncology. First, because they are highly prevalent. Secondly, because they are very costly. In this panel here, you can see the most popular oncology drugs that has been used nowadays in the clinics. Underneath each circle, or each cheese, I don't know what you call it, you see the costs of treating non-responders of those therapeutics. We're talking about billions in several cancers. So let's talk about some recent advances in biomarkers and also, of course, in therapeutics, because when you have a biomarker, you will have, I would say, a new regimen or a new therapy regimen, not necessarily a new therapeutic, but a new type of regimen. But for that, let's go back 10 years. More than 10 years, because 10 years was where a good publication was done by Robert Weinberg. And let's see what makes cancer cell cancerous. So basically, what makes a cancer cell so different than the normal one? In 2000, Robert Weinberg published this paper. I believe that some of you already seen this image a lot of times. The paper was published on Cell, and it calls the hallmarks of cancer. It was not in 2000 that we discovered the hallmarks of cancer, of course, it was much before that. But he made this very nice review about the hallmarks of cancer, where he simplified with this graphic saying that cancer cells have these 6 characteristics that you see there. Self-sufficiency in growth signals and blah, 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 blah. So these are the features that makes cancer cells different from the normal ones. And of course, it's obvious that the current target, these are the current target features that is used for drug development. So drug development nowadays are targeting most of these features to beat cancer cells, to kill cancer cells. And in fact, as you can see, this is some of the most common oncology drugs, and all of them are around the same proteins and around the same targets and around the same pathways. So they are targeting tyrosine kinase receptors on the external part of the surface of the cells, tyrosine kinase pathways that activate cascade of signaling to the nucleus and will activate oncogenes, invasion genes, metastasis genes, survival genes, apoptosis genes, inhibiting apoptosis. So all these kinases and all these signal transduction pathways are related in some way with survival of the cancer cells. So that's why the drugs are built for that specific genes and not for the others. However, in 2011, the same Robert Weinberg published a revisited review of its own paper, where he makes a summary of what we did in the last 10 years in science, and discovered and had to the same characteristics that we see before, he had another 4. That are these ones. Tumor cells deregulates cellular energetics, that's a new feature. Tumor cells avoid immune destruction. They are related with tumor-promoting inflammation, and they harbor genomic instability and mutations. That's we were much time before, but when he said that, he wants to include DNA repair instability. So what we're expecting is that the new biomarkers and the new therapeutics will start made to target these new features. In fact, in the last 5 to 7 years, we are starting to see by institutions, research institutions, and also by pharma, a great investment in new kind of drugs that not are related with tyrosine kinase and signal transduction pathways. So we start building aerobic glycolysis inhibitors, PARP inhibitors. PARP are genes related with DNA repair, selective anti-inflammatory drugs, and also immune-activating drugs. To be honest with you, I think immune-activating drugs will be the future for a great part of cancers. It's absolutely amazing. I will talk about this further on. Actually, this is my first example. I just brought 2 examples so you to see. One of the very promising therapeutics that has been in clinical trials, in a very good position now in clinical trials, are PARP inhibitors. That has been used in gynecologic cancers like breast and ovarian cancers. Basically, the results are very promising, especially in BRCA1/2 associated cancer. You know that some of the cancers harbors BRCA1 mutation. So because they don't have BRCA1 mutation, that is a DNA repair gene, so during the tumor progression, they will lose this gene that repairs the damage in DNA, so they don't have anymore this homologous recombination system. These cells become much more sensitive to base excision repair genes, and one of the genes is PARP. So if you inhibit PARP, the tumor cell is no longer able to repair its own DNA, and it enters into apoptosis. So you are killing the cell because the cells are not able to reconstruct their own DNA. The second example is the example that I really like, is the immune activating therapy. This case is anti-CTLA-4. CTLA-4 is produced and is expressed in the surface of T cells, cytotoxic T cells, and tumor cells use a strategy that is stimulating CTLA-4 on the T cells, and then they turn off the engine. So T cells stop attacking tumor cells because they are stimulated on CTLA-4. But some researchers from UK and I think it's from France as well, together in a consortium with some groups from the United States, discovered that if they kidnap CTLA-4 and block CTLA-4 with a monoclonal antibody, they can restore T-cell proliferation, differentiation, and survival, and maintain the activity of T-cells. And these T-cells, our own immune system, will still beating or still fighting cancer cells. So this is absolutely fantastic because we are using our own biological system. We are just boosting our own and improving the ability of our own immune system to kill cancer cells. I think this will be really good in the future. Biomarkers first, second, omics platforms. That is also related to biomarkers, because omics platforms, especially the next generation sequencing and whole genome sequencing, whole exome, PCR amplification, transcriptome RNA, exon capture, all of these amazing technologies that we have available today will improve a lot the biomarker identification. I won't take too long on this slide. 3, biobanks. No one can do personalized medicine or translation research without biobanks. Actually, biobanks is the bridge between translation research and personalized medicine. There's a huge number of publications coming in the last few years about how to govern a biobank, how to form a biobank, ethics issues, a lot of things that have been published in biobanks. Because biobanks are really, really important. They constitute a new generation of new knowledge in biomedicine. So they're important for the evaluation of new predictive factors. They're important for search of new therapeutic targets, for validation of new diagnostic markers, and for the elucidation of new molecular pathways. So basically, biobanks, a very well-characterized biobank will open the window for validate what we are doing inside the lab. Otherwise, it's not possible to move forward. So that is the translation of new biomarkers into clinical practice. So let's see how how academia is dealing with the new world of translation and personalized medicine. This macroeconomic health research global environment that we are living makes translation research and personalized medicine an increased focus of funding agencies. And we know that at least in countries like in Portugal and not many others, there's not too much money to do research if it is not applied to something. And it also makes it possible to expand our research funding, making science more eligible for new type of grants. And it's a very prestigious area of interest for academia. So, but who or what is feeding this fish tank, where clinicians are, scientists, doctors, patients, everyone? From one side, directly from pharma, most of the big pharma started in the last 5 years, start having on their websites a part that says investigator-initiated studies. Why? Because pharma is still seeking a lot of new ideas coming from scientists. So pharma also is paying for very good and very high quality research and innovation done in universities. The European Commission is also supplying a lot of and sponsoring a lot of research, and both through public partnerships, public-private partnerships, as is the case of IMI, Horizon 2020, as you know. There's a lot of calls that is devoted to translation research and personalized medicine, European infrastructure for translation medicine that works as a platform that manages and regulates scientific contracts between pharma and academia. But I want to give more emphasis in IMI. IMI is the largest public-private initiative in Europe, aiming to speed up the development of better and safer medicines for patients. Basically, IMI supports collaborative research projects and build networks of industrial and academia experts in Europe. And it's very important because IMI is constituted by European Federation of Pharma Institutes Associations. So all the projects that are reviewed inside IMI are approved if they are really good. At Ipatimup, that is the research center that I'm working for in the last 14 years. Ipatimup is a cancer research center. It's a comprehensive cancer research center because it's connected with a lot of hospitals and especially with the oncology hospital here in Porto. This proximity gives us a lot of clinical input and capacity to give support to clinical trials. I should say that is the biggest and largest oncology research center in Portugal. We have a lot of research model and facilities. We are mostly well known internationally by our research, especially in gastric, colorectal, and thyroid cancer. We also have our models, but we have very good facilities that now are moving forward towards personalized medicine, next generation sequence, proteomics, animal facilities, and the very well developed genetic diagnosis and pathologies that work, at least I would say it's with 85% of pharma companies. And we have also groups dedicated for new therapies. In terms of research areas, we have research groups that are more devoted to basic research. It's very important that all the institutes that want to start making translation research or personalized medicine do not forget basic research, because this is what makes them high standard. Otherwise, no one will seek these people. We have groups that are more devoted in translation research projects that are more adapted with interest in pharma, and projects that are really in personalized medicine, that are studying genetic heterogeneity of cancers, developing new biomarkers, developing new targets, seeing the genomic landscape of different tumors of the same type, to see why they are different, and to start developing new strategies to beat cancer. So recently we create the Ipatimup Innovation and Translation Research with the aim to promote the interface between the industrial communities, in academic research teams and clinicians, and to reinforce the role of Ipatimup as an R&D partner of excellence with international pharma and biotech companies. And the other side, from the side of innovation, we encourage the foundation of growth and new profitable spin-off among our researchers. As well, we help them to protect their intellectual property from the knowledge that they are generating on a daily basis. We have a lot of projects on the innovation settings, but I cannot go through all of these. Some of these are highly confidential. I will just give you an examples of the type of projects that we have with Big Pharma at this moment. Basically, most of them are biomarker-based projects. I would say de-risking projects, as I explained before. So we collaborate with a lot of pharmaceutical companies, with the global entities, with global parts. One of the example of projects that we had recently was with a Portuguese biotech recently created, was Coimbra Genomics, and this is a partnership with China, with BGI. BGI is Beijing Genomic Institute. It's the biggest center in the world with the largest capacity to deep sequencing. And basically this project aims to unreveal the genetic basis of gastric cancer in order to develop clinical decision support software tools. So I would say that this is a really personalized cancer medicine project. The second example that I bring to you, and we have two independent projects running at this stage. aims to study the cell surface protein modifications to understand and to improve monoclonal antibody-based therapy for cancer and also for inflammatory disease. Basically to study why certain drugs that are in the market on these companies, although they are eligible for, let's say, 60% of patients, why they are working just 15% of these 60. So there's a problem on the surface, on the contact, on the matching of antibody with the surface, and we are trying to improve the number of patients that they can treat by giving someone is online there. So basically this project aims to identify specific biomarkers to help stratify patients and select the ones that will better respond to treatment. The third example that I give to you, it's a more basic, not so translation. It aims to identify newly oncogenic signaling pathways in medullary thyroid carcinoma. And finally, the fourth example, it's a more broad project that includes several research groups and also includes the genetic unit that we have, a molecular genetic unit. And this unit makes a lot of partnerships with pharma industry in order to make screening of tumor-associated genetic alterations for therapeutic decision directly on the clinic. And we are doing this on a daily basis. And I should say that this unit is one of the few that is certified to do the development and validation of next-generation sequencing-based strategy for diagnosis of hereditary breast cancer and ovarian, BRCA1 and 2 mutation, using this technology that is Young-Torrent technology, the next-generation sequencing. So I'm finishing my presentation. I just want to let you this slide just to see how fast are we doing in the last years. And how has been the changing in the way that we are looking for medicine. I would say that translation research and personalized medicine will definitely reshape the way we see medicine today. And the one that will benefit this is you. So we won't cure cancer in the next 50 years, but we definitely will be able to make cancer a chronic illness in the very, very near future. As you can see, in the last 20 years, the last 1,000 years actually, we were treating and diagnosing diseases based on the anatomy, based on one we could see, we could smell, taste it, pulp it. So we were in the anatomical setting era. So in the last 100 years, we moved forward for the cellular level. Now we start diagnosing and treating people based on increased knowledge on their biochemistry, on their cellular process, on their pathways. Nowadays, we are in the molecular and gene level. We treat and diagnose people based on their molecular and gene profile, in their deep biology, knowing as a signature, as a molecular signature that everyone has in their own diseases. I believe that in the very near future, with all the data mining and test mining that Professor Zelwij was talking today, we will be able to introduce all these characteristics from a single patient together with their personal characteristics and all the screening of molecular and genetic profile of that patient and ask to the computers which will be the best therapeutics to this patient, because the computer will already know all the results from the clinical trials and will join all together this information. So we will living in the prediction era that it was made by IT, information technology. That's why it's so important in the future. So the question is, are we winning all this evolving war on cancer? Professor José Baselga published this very recently, saying that could this mark a turning point in this war of cancer? The emerging therapies, the new biomarkers, the personalized medicine? I would say yes, definitely yes. As I said, we won't cure cancer in 50 years, but we can make cancer a chronic illness. So everyone will, at least for most of the cancers. So just to let you with this message, this is a time, your time, is an exceptional promise and a great reward for all cancer researchers and pharma researchers as well. So it's come not too soon for patients with cancer, that the ones that are in need. Thank you very much.
I don't, okay, it's working. Well, thank you very much for a very interesting and promising talk for the future. I would like to know if anyone would like to ask some questions, because Dr. André will probably not be able to stay longer with us for this afternoon. So if you have any burning questions, comments. No questions and comments? Still digesting the information after the lunch?
Was clear enough.
Maybe I can ask a question.
Yeah, sure. I will make my best to be at the round table, but yeah, go for it.
No, but I would like to know, you talked about specific projects that the Patimup is developing, and you talked about working with the pharmaceutical industry, specifically on some projects. My question is, in terms of but we are hearing more and more about the importance of governments themselves having money or financing this type of research, because some of the research may present a conflict of interest from the industry side. Do you see any conflict of interest coming up in this type of research for the funders of the research? Because nowadays
for the classical funders.
Yes.
No, there's no conflict of interest at all. Basically, for institutions that are the classical funders, for them it's great that we are looking for alternatives source of funding. There's no conflict of interest from the side of pharma, apart from the ones that are related with confidentiality. In terms of national policy of science, this is also seen by the government as a very good way to make our own research made in Portugal much more prestigious than before. So there's no conflict of interest in terms of, and this increases our value in terms of intellectual property, and this is a very good, how do you say, asset for the future. So there's no conflict of interest anyway.
Thank you. Any questions from the audience? Yes, please. There's a microphone coming. Maybe it's better. It's a very big room.
Thank you. Thank you for your presentation. And I have a question. I'm Ocean from Taiwan. And sometimes in this kind of research, it seems a little difficult in like design the research, because sometimes we have difficulty in decide what biomarkers to look in to be more specific on the research. So do you have any suggestion on how to design this kind of research or experiments? Or do you have any experiments like using the tools which the bioinformatics tools which Professor Jose you introduced in the morning?
Yeah, that's a very good question. Of course, that's make part of my daily basis is to first of all try to create a bottleneck approach to the problem or to the question we are trying to address with each company. So the companies doesn't come to us and say, just solve this problem. No, the problem is very specific and includes and involves one or two or three entities, molecular entities or pathways. It does not go too much from there. So we have facilities in-house, that help us, like proteomics and bioinformatics, that helps to narrow the spectrum where we are looking on. Of course we have, and that's crucial. But sometimes it's not very, we don't recur too much to this before we start thinking about the project. Because what we do in a daily basis is to be very updated with the clinical trials. So as a scientist, as an oncology scientist, when we start studying that specific clinical trial and try to understand the biology of that drug and the biology of the type of patients that are the target of that drug, we can start narrowing and making a bottleneck approach to what we want to start thinking in terms of biomarkers. So basically what I want to say is that when we go for a project and we start designing a project for Novartis or Bayer or Merck Serono, we are not doing alone. That's the first thing. We designed the project together with the scientists from the pharma. They come to us or we go there, we sit at the same table, we talk at the same language, and in confidentiality, we try to understand how we can address that specific problem. So what I want to tell you is that when we reach this point, we are not looking to the forest. We are just looking to 1 or 3 trees. We are not looking that much wide spectrum. If so, we have the specific tools to narrow this biomarker platform identification.
Thank you. Any further questions? Doesn't look like it. So thank you very much again for your presentation. Okay, and it now seems that I'm presenting on translating research findings into pharmacy practice and the role of education. Okay, so I think I will be a little bit of an outlier here because I'm not a scientist myself, or at least I am a pharmaceutical scientist, but mostly what I do is I practice pharmacy in community pharmacy, which is pretty much bedside aspect or bedside side of the matter. So I will talk a little bit about, for me as a pharmacist and as a pharmacy practitioner, how do I see the importance of translating research findings into pharmacy practice and the role of education in this regard. So I know that you've talked a lot about the issue at hand. So as you know, or should know by now, clinical and translational science is a discipline that is focused on speeding the movement or translation of scientific discoveries into effective, accessible medical treatments that improve health. Of course, this sentence can be read in very different ways. And in fact, if you look at this image, which I found on the internet and I consider very interesting, is that when you're looking at translations and when you're looking at different steps of what happens with research, there's a lot of steps. There's not only science to practice. So there's the issue of having a molecule and then working on the molecule or the molecules to find one that is safe and that is effective. But then there's the issue of taking that molecule into the community, into the individual patients, and understand how it works, if it works, and how we can expedite not only its commercialization, but also the important impact on health that every molecule can have, has the potential to have, but sometimes in practice that does not happen. So I will try to talk a little bit on my perspective or pharmacy practice perspective, how is this important. So you've seen this image today, and basically it just relates to the fact that between what is found in terms of the lab and in terms of science and what happens in terms of practice and when solutions are really used to save lives, there's a gap in the middle and people are simply dying whilst we do not implement what we already know in practice. Okay, so we start at the bench, but then what does the pharmacist do, and what do we need, or how does the public perceive us when we are talking about pharmacy practice? So this lady here, she was wondering, what does the pharmacist do with her prescription? So the pharmacist ensures that the medicine, the dose and the instructions are safe and effective, that the medicine will not react with anything else she's taking, that the pharmacist also warns of any significant side effects, that the doctor is contacted if the lady has any concerns, and the individual medicine's record is updated and maintained. Of course, we can do all this just with the knowledge we have on the drug itself, and basically what was discovered in terms of clinical trials. But we can also nowadays, like you've seen this throughout this day, we can now use different data rather than the one that we obtain in normal, regular clinical trials. We can use different data, and we should be producing new evidence in order to connect not only what the medicine has the potential to do in terms of side effects, in terms of efficacy, in terms of interactions, but in that specific case, with this lady in particular, knowing what her genome looks like, what will that medicine produce in terms of, again, side effects, interactions, etc. In different people, medicines can work in different ways, and that's what we have seen today. And those of you that have attended the workshop I've led, I think it was on Saturday, have already known this image. And this is basically, this tries to depict what will be the health solutions in the future that the pharmacist must be ready to provide. So not only the active substance itself, which is in the middle, and of course we will continue to be, as pharmacists, the experts on medication, but we will also have to add all these interventions and counseling, or at least recommend other healthcare professionals that can provide this type of interventions and counseling in order to maximize the benefits of therapeutics, be it the efficacy and the safety itself. So, for instance, when you get a diabetes patient, it's not only important that he is medicated with the right medicine, be it metformin or insulin or any other medication for diabetes, but also that he has adequate advice on his food regimen, practices exercise, has interventions to maximize his compliance to the medication, which we know usually does not happen in real life. And again, we should also add to this solution and to the molecule all this knowledge that comes from translational science research. And in the future, maybe we will have this kind of medicine being dispensed in our pharmacies. Nowadays, we have these medication being dispensed at hospital level, and most of this research is already applied in clinical settings. Most of the times, though, in pilot studies or not. We can say that it's not completely disseminated, and it's not being used at the population level. But we will eventually have a time when every medication we can have, we can test if that medication will be effective and safe to use in that particular individual. It's not that far away. In fact, if you go to the internet and do a little bit of research, you can already find some websites where you are able to buy your own DNA or personal genome service. So this is a website I found, so Get to Know Your DNA. All it takes is a little bit of spit. So it's not complicated. People can get access to it. But what do we do with the results afterwards? And this is one of the major challenges for healthcare professionals, is what we will then do with the information that we gather. And we have talked about, and I think maybe most of the time when we are talking about translational science and we are talking about translational health, we are talking about personalized medicine, and we are talking specifically in pharmacy, we are talking about pharmacogenomics. But the truth of the matter is that if we take the definition of translational medicine and we look at, again, what it aims to achieve is effectiveness and the best use of medication, we are also not having the maximum benefit from the medication portfolio we already have in the community. And this was a study done by IMS, an international study, and basically it aimed to highlight or to advocate for a more responsible use of medicines to improve health outcomes and reduce costs worldwide. And they begin by talking about why it matters and why should we care about advocating for responsible medicine use. And what they say is that medicines are an integral part of healthcare. However, policies for medicine are often used isolated in the intensifying efforts to contain costs across the overall health system. So we sometimes are very worried or too concerned about the gaps that exist in research, but are we also worried about what happens in the implementation phase? So there is an increased availability of necessary medicines, there is expanded access by patients, which equals higher volumes. So the global volumes of medicines have risen substantially over the last 10 years, and you can see some of the percentage presented here in this slide. But what can be improved? And what they argue is that improvement can be done at various levels of implementation and translating what we have found in terms of science into what has to work in practice. So there's the issue of non-adherence to medicines, there's the untimely medicine use, antibiotic misuse and or overuse, there are medication errors, suboptimal generic use, and mismanaged polypharmacy. And when we count all of this, what's at stake in terms of economic value, nearly $500 billion is the avoidable cost opportunity worldwide that could have been realized by these 6 different levers in 2011. So there's a lot to do also with the medication portfolio we already have in terms of implementation and making sure that they actually work. So when they listed the 5 key potential recommendations, the first one was to support a greater role of pharmacists to own medicines management. And when we are talking again about medicines management, we are talking about all kinds of different different drugs, but also personalizing our interventions to what we know that individual might experience due to his specific characteristics. What can we do as pharmacists? And probably, as Dr. André has highlighted, nowadays it's not as easy to understand what a certain person needs in terms of pharmacotherapy, because it's no longer what we see, it's no longer what we taste, it's no longer what we smell, it's things that we have to study, it's things that we have to discover. So maybe we will need to have a different approach to understanding what kind of needs that specific individual has. And just because it's not something that we can easily see, it doesn't mean that we should not do something about it. And this is basically the important thing, is that although it's more difficult in terms of personalizing our intervention, we have no excuses not to do so due to the scientific advances that were already presented to you here today. When we're looking at this concept of translational research, you've already talked about the importance of translating basic research to bedside implementation, but then there's also introducing it to the clinicians, but there's also this important step about policy and practice, translating it into policy and translating it into practice itself. And so practice-based research is also one of the areas that has to be fostered inside our institutions, academic institutions, but also when we are providing care in the community, we have to find a system whereby we can feed all the information about the use of medicines into the science part, into academia, all these institutes that are looking at the development of medicines and and translational science, because without this information they cannot feed on their research questions and move towards having more effective and safer medicines, not because the medicine itself is safe and effective, but because its use by the patient is safe and effective. So we've already talked about pharmacogenetics, and one of the things I would like to highlight when I'm talking about the role of education in this subject is that in pharmacy, most of the universities and most of the faculties that have integrated the concept of translational science in their curricula, they have mostly gone into pharmacogenetics and pharmacogenomics and not really on the whole spectrum of the problem. And of course, it will be possible to have a patient group with the same diagnosis, with the same prescription, and through studying all these variabilities, you will find that in some groups the drug is toxic and is not beneficial, so you should definitely not use the drug in that group of people. But there will be a group of people where the drug will be toxic and beneficial, so this will be something that can help us make our decisions. Like I said, on the translational continuum, though, there's not only one valley of death. So I've showed you the picture that came up in the Nature journal in 2008, and it showed one gap between research or bench and bedside, but in fact, researchers have pointed out that there are at least two different valleys. So there's the first one, which is a decreased capacity, which translates or depicts a decreased capacity to translate research results to the next stage, affecting first, and this is the first one, our ability to commercialize research results. But then there's also a second one on our effectiveness at moving results into clinical practice. And these are two different valleys. And when we are talking about education, we have to make sure that we provide the necessary competencies and skills to pharmacists to be able to work not only on bridging the gap between basic biomedical research and clinical science and knowledge, but also its application into practice and health decision-making. And this is something that the Institute on Translational Health Sciences is already working on, is that they identified different stages, let's say so, and then they have people working in these different stages to make sure that in the continuum of care and in the continuum of research to care, these valleys are sort of flattened. So since the identification of problems, opportunities, and approaches to the discovery or foundational research, then we have the health application to assess efficacy, then health practice, the science of dissemination and implementation, and then you have also the evaluation of the health impact on real-world populations. So again, if we do not have practitioners feeding this data into this stage of research, those that are working in the T4 do not have sufficient data in order to be able to advance their research questions. And basically, this sort of depicts a structure within the Institute for Translational Medicine in terms of organization and capabilities, where you really see the importance that this stage has for translational research and translational medicine. The community engagement cluster that you see in pink, so they have a connection and participation from community practitioners, and then they also have a population sciences cluster where they develop population-based health studies and where they also study or include in their research social, environmental, and non-biological determinants of healthcare. Okay, so in this ecosystem, what do we as pharmacists need in terms of evolving our undergraduate curriculum? Well, I think that we are in the very early stages of understanding this. And I'm a little bit worried that the time that we are taking to introduce this in our curricula is longer than the time that we were already complaining about the research results and the implementation in practice. So we are taking too much time to implement these changes in our pharmacy curriculum. Because I believe, and I'm sure that you will agree with me, that our role as only translating doctors' prescriptions is over. This does not add value to health systems. And I'm sure that you also agree with me that continuing education is important. I must tell you, I'm not that old, and when I was in the university, when I attended pharmacy course, we did not talk about pharmacogenomics at all. So this is something that is advancing really fast, and us as pharmacists, and one of the significant roles of education in this regard is that we have to make sure that we understand that continuing education development is essential for us to be able and capable in this evolving healthcare system. So like I said, I believe that we are in the very early stages of understanding what we have to do in terms of education and what the role of education is. I do, particularly in terms of undergraduate curricula for pharmacy, I do believe that we have to develop our capacities and skills in terms of formulating research questions, be it at the more science-based research, but also on the implementation research. The FIP Education Initiative is just doing that. I know that yesterday you had the opportunity to contact with several experts that are involved in the FIPED Education Initiative. We are now just working on a new domain for the FIPED Education Initiative that includes the FIPS interest group on translational medicine and personalized medicine. to be able to understand what is needed in terms of education so that we can also add it to the core competencies that pharmacists will need to in order to make valuable contributions to this theme in the future. So, and because if you're not part of the solution, you're part of the precipitate, a little bit a joke on the pharmacist side, but I am sure that we will be able to contribute if we are able to do what we are complaining or what we have said that others are not able to do, which is include these disciplines in our pharmacy undergraduate curricula. Thank you very much. I will just ask about further organization. Do you have any questions? Yes. Okay.
Hi, my name is Hyunji from South Korea. About the valley that you described, there were 3 parts, but like the meaning valley doesn't mean like there are obstacles to move on. And if that means obstacles, then could you specify what are the obstacles? Thank you.
Sorry, in which one?
The valley, 2 valleys that you described.
Okay. In this translation continuum, right?
Yeah.
Let's see if I can show. So one of the values that we have discussed, and the one that was basically presented by Dr. André first, was there's a gap or a value between what is basic biomedical research and then the commerce to be able to commercialize the product itself. So sometimes due to different barriers in terms of research, there's one molecule or a specific type of product that the pharmaceutical industry is having difficulty advancing through the different clinical stages of development. So basically, a lot of translational medicine institutes are trying to, in a way, help to address these barriers and advance medication to its distribution stage, so commercialization. The second value that has been presented by these researchers is the one between clinical science and knowledge. So we are able to commercialize, but then our effectiveness at moving results into clinical practice. The fact that the medication is used for the purpose that has been advanced. And sometimes it just means that, for instance, governments are not uptaking the medication and not including them in their formularies, or they're not included in clinical pathways, or simply clinical practitioners have not seen in which type of patients they are to be used. So you need research to bridge those 2 different valleys. So the one that allows you to reach the final end stage of commercialization of a drug, but then another one that allows you to really obtain the maximum benefit and efficiency from the drug that has been commercialized. Any further questions? Yes.
I can yell. Oh, okay. Good afternoon. My name is Brittany Monty. I am from the USA, and my question is about the pharmacogenomics portion. You said one of the challenges was when we do get all of this genomic information, there's an issue on what to do with it. So my question is, in the future, do you perceive how we use the genomic information is a patient gets prescribed a medication, and then we check for mutation. We check for mutations within their genomics, or do you think before, we just view a patient, review their genome, and we just try to avoid certain classes of medication that they may be subtherapeutic or toxic to?
Well, there are different types. I'm not an expert on this field, but I know there are a lot of results that come forth from this type of research. So it may be regarding the way that you metabolize the different drugs. So for some people, it just means that the doses have to be adjusted according to the metabolism of that specific person, because the same dose that is used on a certain person can be suboptimal in terms to achieve efficacy, and at the same time, the same amount of drug in another person can be toxic. So it depends on your ability to metabolize the drug. And of course, this also depends on the drug, whether the active substance is active as an active substance or as a metabolite. So it has many different stages, and the conclusions may be completely different. What I meant with also with what we can do with the information, and I think that we will have there is information such as the adjustment of doses that will be quite straightforward. So we will probably be able to use different algorithms in order to calculate the specific dose that has to be given to a certain person in order to achieve efficacy, but at the same time be safe. But there are other types of conclusions that we can take from genome testing and or these kind of processes that I believe are a little bit more difficult to use. I attended this symposium where they were presenting the fact that, for instance, for a certain group, for when you test women, some women are some women will respond better to certain cancer drugs than others, and there are even some subgroups of women that respond that have a very decreased response level to certain cancer drugs, and who actually can have more harm due to the toxic effects. And for instance, but even in those women that do have that type of genotype, the issue is that even for some, the medication can be effective. So there's always a percentage associated with it. And I think that it will present ethical challenges, and I think that ethics will become increasingly important as a discipline in our undergraduate curricula, because who will then decide whether the percentage is relevant or not if you give it to one woman and will not give it to another? It's a very complex decision to take. Will you work on statistics? Will you rely on those kind of statistics, or will you look at this issue person by person? It's very tricky what you can do with these results. Some of the applications are very straightforward. Some of them present real ethical challenges, I believe, for all health professionals involved. Okay.
Hi. So I think Mike is. So it's a very nice talk on the various aspects of translational medicine. So I just have few comments. As you have mentioned, the various stages of the translational medicine process from T1 to T4, and you gave the reference of NIH, and you gave the reference of 2008. So I just want to add that in NIH, now there is establishment of NCATs. So NCAT has is like a kind of platform for all the CTSAs and all kind of these initiatives. And that concept of T1 to T4 is not the compulsory kind of the understanding of NIH. So it's just a kind of the belief as like the various kind of concepts. And the second thing about the, so you have mentioned about the valleys. So it's also only a one concept. So there is also, concept in which they say there's only the one valley of death. In regard to translational medicine, so there are the various kind of theories, and so there are the various kinds of understanding, and each institutions and departments, they have, tailored the concepts and the kind of the definitions according to their requirements and according to their needs. So it's only I wanted to add. And you are like,
it's truly an evolving discipline. From what I've researched, like I said, I'm not an expert in the field of translational medicine, and translational sciences. So from the practice side, what I see is what I get as a practitioner to work, and what I still see is some delay in what I often see for instance, in congresses of what is being discovered and the actual responses or the actual portfolio of drugs and interventions that I have available right now in practice. So I believe that education will play a very significant role in adding this awareness for me as a practitioner, for instance, but also, like I said, I think the most important message, at least for me here in terms of education, is that we cannot, if we are arguing and if we are complaining as practitioners that we do not have access to the most recent available data about what is in fact effective and what exists in what is happening in the research field. But again, we are not able to introduce that early on in our undergraduate curricula, that we ourselves are causing a delay in the process. And we should not only blame others for not having that available, but also look a little bit on the inside and see that we also have some responsibility in moving things forward, in introducing this type of issues and research, etc., in our undergraduate curricula.
Yeah, it's true. Main problem is that so there is a demand or the requirement to have the standardization or the consensus on the various aspects of the translational medicine, including education and the training, including the definition also. And regarding the various educational programs, so there are still, the many institutions, especially in UK, Australia, and so on. So there are already master's program and so on. But still, there is a need to standardize the curricula.
Yeah. Thank you.
Hi, I'm Acacia, BPSA UK. You mentioned that it may take too long to integrate the concept of translation medicine into our undergraduate curricula. So how would you suggest that the younger generation of students educate themselves on this growing concept in the current present? Yeah. Thanks.
Well, that's a difficult question because I myself, when I was looking for the information, we have all these institutes, and we have different graduate courses and different education opportunities for translational medicines in institutes that have developed this kind of expertise. But when I was looking into specific pharmacy curricula that had included translational research and translational sciences in their undergraduate curricula, I couldn't find any information. So basically what I would say is that there are courses available for people that have as a basis an undergraduate, a graduate curricula that is health science related, but then you need to engage with these institutes for translational medicine and translational science in order to gain more expertise on these issues. It's something that is, for the moment, I see it very separate from the undergraduate curricula that exists for pharmacy. And I believe that, in a way, also for other healthcare professionals. So it has to be on top of education. I don't know if you have any...
Yeah, actually you are right. So as you said, that at the level of studies under like if you are just a student of pharmacy or the veterinary medicine or so on, so at this level, I would say there is no need to go for translational medicine. It's a pure, a new, specialty or the field, and at the level of the post, graduation, so there are specialized master program, even MPhil and the PhD programs are in the market, I would say so-called. Yeah, but you are right that at the level of undergraduate, I don't see any kind of opportunities also, or there's a scope of translational medicine.
Yes, and I do think that we cannot put everything in our graduate curricula, but I think that we at least should have a head start or a discipline that may be able to open our eyes for this issue and then pursue further education opportunities such as master's courses, etc., if we are going into that field, but at least have some perspective in the faculty already. Okay, so I think we will move to the coffee break. Oh, there's a question over there. Sorry. But we will have a round table afterwards, so with the real experts on the matter.
Thank you for your presentation. I'm Jegne and I'm from Slovenia. If I'm not mistaken, you mentioned in your presentation that we need more practice-based research. Now, this may be a little bit of an offbeat question, but since you're a community pharmacist, I'd like to know what's your opinion. Could or should we do more of this practice-based research in community pharmacies? The reason I ask this is because I don't see a lot of that happening in my country, but I don't know what's the situation around the world. Thank you.
Well, I think that overall the situation is not very different from your country. So there is very few practice research centers that I know of. There are a few that are doing very interesting work, and some initiatives that are being undertaken, and some practice research that is going on because national or regional organizations thought that it was important for their advocacy work, and therefore have developed different projects. For instance, the foundation of the APHA Foundation in the US, they have undertaken different research projects. For instance, one that's very well known, the Asheville Project, which was a project that they studied the effect on population in a certain region on diabetes patients from that region of pharmacist interventions to those patients. And they did a follow-up study on those patients following the pharmacist intervention and drug management, and then they did the cost-effectiveness analysis of that intervention. And this is one of the few big studies looking into interventions in the community. And the second biggest problem I see with practice research intervention is that most studies that are undertaken and that have been undertaken within institutions such as like the Pharmaceutical Care Network Europe, for instance, what they did was they developed a project and an intervention, and then they studied the intervention. But at the end of the study, the intervention stopped. So it was not assimilated and it was not disseminated. It was not translated into practice afterwards. So we have very interesting data about the effectiveness of pharmacist intervention in practice from those studies. But after the study, the intervention is dead. So that's another challenge for practice-based research. I do believe that it will be increasingly important. You may have heard that governments are nowadays increasingly discussing about having what they call risk-sharing policies, which means that, for instance, when pharmaceutical companies want to introduce a new drug in the market, and usually they enter the market with their data on effectiveness and safety, and then they negotiate for a price with the government. Nowadays, what the governments are saying is that, okay, I will allow your drug to enter the market because it's effective, because it's safe. But now I want to see how it really, from your clinical studies, but now I really want to see what are the results in the real population, and if the results in real populations in the real population are not as you are advocating they are or advertising they are, then I want you to reimburse me back for those drugs that I'm paying for. So increasingly, it's becoming important to have data from real population, and where the real population is in the community, in hospitals. So I believe that the governments and health insurance companies will have to rely more on the involvement of pharmacists, doctors, etc., that are at the practice level in order to gather that data and to study those, if the drug is being or not effective in the community. And at the same time, I believe that the pharmaceutical industry will also have an extra incentive to work with pharmacists and to work with doctors to make sure that the drug is safe and effective in the real population. So I think it's a new paradigm that we will observe in the next few years. Okay, so we will move to the coffee break and we will reconvene at 5:30. Thank you.
Dois. Dois. Som, dois. Som, dois. Dois.
Hello. Hello, everyone. Can we start to go back to our seats so we can start the next session in 5 minutes? Thank you. Hello, everyone. Can we invite you back to your seat so we can start the discussion session? Immediately, yeah.
Okay. Good afternoon again. May I ask you please find your seats? We will start this last portion of the symposium. Okay, so thank you very much for coming back from your coffee break and participate in this roundtable discussion. The roundtable discussion topic is on the future of translational research. I will add translational research and medicine. And we will be talking about challenges and opportunities for the future. And we would really like to hear your thoughts about what do you perceive are the challenges and opportunities in this field after having heard from what it is and what it means and what it can mean for pharmacists which are working in different fields of practice. To start off the discussion, I will first ask our previous speakers. Oh, great. We will first ask our previous speakers to name one challenge and one opportunity, or if they believe that challenges are opportunities, maybe they can just contribute with a couple of opportunities, and we will then move to involving you in the discussion. So maybe we can start with Professor Amir Shahzad.
To start about the future and perspective of translational medicine and the potential opportunities in the field. I think the most important kind of challenge in the field of translational medicine is itself to make a standardized kind of the definition of translational medicine. As I was speaking in my talk, that all over the world, so there are so many institutions and departments and the specialities. So they have their own concept of translational medicine, and they have the tailored kind of concept and the definition of translational medicine. According to some, so they have like a concept that it's a two-way kind of process involving the bench and bedside, and according to others, so they say that is a translation of the, kind of lab investigations and the findings into the clinical applications. the most important, challenge in the field of translational medicine is to standardize concept and the definition. And along with that, so there are so many like kind of challenges or like aspects of the TM that are still, the need, a kind of consideration to have a consensus and standardization.
Thank you. Professor José Luís Oliveira, any thoughts on challenges and opportunities for the future?
Okay. Well, lots of opportunities, lots of challenge. Maybe I will focus, being an IT person, I will focus on information management. We will have increasingly more data to analyze, and this will raise lots of problems. IT problem, because we need to have good solutions to handle this data. Brain, we are part of the solutions. Software will not solve everything, so we need to be experts on the field to understand if the algorithms, the data analysis that comes from the raw data means something. And finally, in the between, we need also to manage the expectation of this research. In several fields in the past, every time it appears a new HIP, there were lots of expectation about the possible outcome of the research. And well, I'm not an expert in the field, in the biomedical field, but my experience the last 10 years shows me that there have been lots of resources invested in this new area, personalized medicine, translational research. But at the end, the new steps that we can give as a result of those research are quite small. And this is not necessarily bad, because this is the way we do small steps in humankind. But we need to understand that there is no big revolution on this new approach. Opportunities, of course, there are lots of opportunities. There are opportunities for IT persons, for physicians, for pharmacologists. And I will comment a bit some discussion in the last section. We will not be able to be the perfect researcher that is able to understand everything about any field. This is not possible. So it will be not possible to integrate every knowledge that you will like to know in your graduate courses. The solution passes again for our ability to work in multidisciplinary teams. And in those teams, there will be persons specialized on one of those particular fields, on IT, on pharmacology, on medicine, on molecular biology, on NGS, whatever. So this is the key for I think.
Thank you very much. Dr. André Albergaria. Same question.
So yes, there's a lot of challenges and opportunities. Actually, some month or a month and a half ago, I was in Berlin for a European conference to decide what will be the rules, European rules for the next calls for the next years until 2020 in terms of personalized medicine. So the output of these meetings were two volumes of this size about opportunities and challenges about personalized medicine. Of course, that we can select, and it's good to be here with different perspectives, but I still think that, of course, I was representing Portugal at that time, so I was biased by our need in certain areas like in clinical trials. But the challenge that I see in the next future for the translation research in personalized medicine will be a better conciliation of patient data. That was actually what Luis was saying. We need the information about the patients stopped to be wrote with a pen by the doctors and started to improve our information technology so everyone can deposit the information about their patients into a system where researchers and clinicians and pharmaceutical industry can have access in a standardized way. And this way we're going to get better design with more rational clinical trials. The good thing of clinical trials, well done, it has a lot of benefits. Clinical trials are important to bring new therapies for patients, but in terms of financial for the countries, especially for a country like Portugal that clinical trials has been devastated in our country, clinical trials bring innovation for our patients and produce some savings also in our health system. The opportunity that I see it's the crisis itself that Europe is facing at this time. So sometimes the crisis pushes forward the need of doing some movements, deep movements. And I think translation research now in personalized medicine is a face of this movement that has been done by the academia and by the doctors and by the industry. So the opportunity here is to bring all together for the same discussion, but to that, these different players should be open to this discussion. They should create specific infrastructures to improve the connection between the patient, the doctor, the scientist, and the pharma. I think we now are living in days that fortunately we have all the conditions to start talking with them in a more productive way. And we hope that the translation research, the product of translation research in the next future will bring better treatments and better solutions for the patient. So infrastructure coming for all this need that is faced by the crisis, clinical trials, and putting everyone talking at the same language.
Yeah, so basically I think that you have all touched similar points that on the one hand we need to have a more standardized concept and definition for us to be talking the same language when we are talking about this topic. In another sense, we have to work in multidisciplinary teams, and we have to work across different settings of practice and information, because we all have useful information for each other, and we have to feed on the processes in order for them to be effective and really be relevant in terms of the data they produce. So I think this is a very interesting challenge, and it builds on the necessity to work collaboratively and to be able to, again, translate what we learn also and put it into practice. Does anyone have any questions, comments? Maybe I have a question for you. Do you feel that that pharmacy curricula is fostering collaborative processes of care and multidisciplinary approaches and facilitating transfer of information? What do you think? Any thoughts on this? Any of you in a university that has multidisciplinary courses? Would you be able to share experience?
Hi, my name is Jessica. I'm from the US. And in my university, we have a course You can talk to the patients with a nurse, nurse student, medical student, and dentist student. We get one patient per semester, and we help them. So we have to create a project so we can see what they need in their daily life. We go to their house and do house visits all together, and then we create the project as a group to see what the patient needs.
Thank you. And would you see that it's easy to then feed the information about the results of that information, of that intervention, into science, so that it can more easily then optimize the medication that are used, for instance?
I think it was easier to talk about the patient, what the patient needed, into like the science background, because things that I missed, the medical student saw, and we were able to talk and see what the patient needed, and the patient was he had easier altogether.
Okay, thank you. Yes, please.
Hi, Acacia, BPSA UK. I do think this is a growing concept. In the UK, different parts they integrate multidisciplinary learning differently in different parts of the UK. Some of them we have workshops where we sit down with medical students, nurses, physios, and then we talk about case studies. But in some places which is integrated better, is that they bring those students, all of those multidisciplinary students, to the hospital, and then they get a real life patient and get them to discuss about their disease and their healthcare and come up with a care plan. So that's really good, and I think this issue is definitely a really big issue. This was the theme of last year's World Health Students Symposium. So that issue is definitely, I think everyone should be aware of, and I think we're definitely building towards integrating that system into our educations, which definitely needs more sort of systemized way, but it's definitely a growing thing. Thank you.
And but is translational research and translational medicine something that is it a concept that is fostered within your pharmacy curricula, for instance?
Yeah, so that's really good. That's why I asked you the question about whether we should integrate that into our education system, because it's not something that I've seen yet done in the UK. I cannot speak for other countries or other parts of the UK, but in my own faculty of pharmacy, we have not seen that. We do learn like pharmacogenetics and things like that, but nothing that relates like translating, research and then into practice. And I think that is really something that, we've spoken about it the whole day today, so I think that's something that all of us should bring back to our own countries and sort of educate all of our members about it, because it's quite a growing issue, I feel. Yeah.
Okay, thank you.
Thank you.
Hi, I'm Helen from New Zealand. Recently in New Zealand, they've brought in a program where 6 different faculties go to a rural area in New Zealand. So there's pharmacy students, medical students, dentistry, physio, nursing, dietetics, and we spend 6 weeks together and we work on cases and projects, and we're able to see the views of different faculties, and we get to go to placements with each other to see what they actually do and what each faculty does, and that way we were able to break the stereotypical views that we have of each faculty and really know what they do and what their job is. And it was really successful, so it's just recently come in, but I think it's going to be developed more.
Okay, thank you. Thank you for sharing that.
Say, Janet, in UK there are a lot of interprofessional learning at university, and I'm sure that it's not a new concept that, students get taught interprofessional learnings at university. But recently, in the for particular regions, they've picked up a new they pair up qualified pharmacists with junior doctors so to educate them better prescribing. So I think maybe introducing interprofessional learning at workplace will be the way to look forward, because we have never experienced that before, and I think that will be useful for all the professional services around the globe.
Okay. Yes.
Yeah, hello, my name is Melanie, a Polish girl from Germany. Yeah, my question, no, it's not a question, it's first a comment and then a question. Yeah, I think we heard, the last days we heard a lot about advancing our competences and widen our horizon, but I think every student agrees here that we have really full curricula, and I don't see that happening until we can study longer, but studying longer is an issue of costs, and I think lots of governments are not willing to pay these costs, and then it's up to companies and other, I think, associations to pay for this cost. And my question is, how do you keep your independence from them? Since I have a feeling that when in the pharmaceutical field, the pharmacists who work right now don't have themselves a really clear view what the future pharmacists should look like in the future. And as students, you just feel disorientated, or I feel disorientated because there's no clear view. So I would like to ask you about this opinion. How do you, keep your, beliefs through, and how do you push them through? And on the same time as keep being independent and from an academic point of view, keep still thinking for yourself and not being biased from too many sides.
Okay, so if I understood correctly, you are asking about the possibilities for pharmacists to get educated in this field. But you were mentioning independent. But I didn't quite understand what was the connection between the studies themselves and the independence of practice. Because I'm sorry, because we can't really hear that well here.
There's a problem with the mic.
Yeah.
Yeah, it's again working. Yeah, my point is that you are supposed to have lots of competences as a student, but I don't see the thing happening without prolonging the studies, because, since it's a matter of costs. And I don't know how it's going to be financed, and my point is just how do you, from the education point of view, how do you keep your independence? And I know there are master's degrees and you can specialize, but if you want to have not too much specific information, you do like a summer school or courses, and you get biased, for example, by companies. And how do you keep your personal independence?
Okay. So what are the opportunities for further education in this field, other than graduate? Yes, maybe Professor Shahzad has some information, and Dr. Andrea Albergaria also about that.
Yeah, so I don't know if you are saying about the same thing. If you are like a student of pharmacy and you are the finished, and so you want to keep your field, but you also want to keep updated about translational medicine as a kind of exit, and to have a kind of edge in your CV and to be updated in the field. So yes, so there are opportunities under our EUSTM. So there is Academy of Translational Medicine, the professionals, and under this academy, so there are the professional, the certification programs are offered under two kind of categories. So category, I mean, one is for those who are graduates in life sciences. And for this, I mean, category, you have to pass a qualifying exam if you want to be certified as a translational medicine professional. And we provide a specified kind of the syllabus, and afterwards you have to be appear on an online examination. So if you would qualify, so you would be awarded the professional, I mean, certification in translational medicine. And under the category 2, if you are already have so much experience and you, I mean, fulfill the criteria set by academy, so you are ultimately qualified. In addition to translational medicine, the kind of expertise is our main tool of translational medicine. For example, the clinical bioinformatics, biomarkers, and public health. So these are also areas where we also offer this professional, the certification programs. I don't know if it's help your, I mean, question.
Yeah, thank you. It's happening.
So I think one of the reasons why it's so difficult, or we didn't reach there yet, in terms of incorporation or incorporating translation research as part of the pharmacy or medicine courses, is due in part due to ambiguous definition that we still have about translation research. As I explained, there's different perspectives, and as Amit said in the beginning, sometimes it's difficult to find a unique definition for translation research. It's not something that is palpable, and it's difficult to build all the contents that incorporate this as a discipline in the courses. What we are doing here in the medical faculty in Porto, I'm an affiliate professor of the faculty, is to start having translation research in personalized medicine optional modules of the course. It is much easier to incorporate this in these courses as additional and optative modules of the course, because you can specialize what you're going to do, and you're not just dissolving the concept in other concepts that is given in these courses. In Europe, you start having some universities, as Amit was saying, in some places that are doing post-graduations and masters on this area of specialization. And the United States are much going further on this setting. There's a very good course in Harvard-MIT Consortium for Translation Research in Medicine every year. It takes it's K1, so I think it's one entire year. They are forming doctors, pharmacists, biologists, and on this type of formation, you don't have just the general concepts about translation research and personalized medicine as we talk today, but you will learn, you will visit the hospitals that have the proper facilities to build translation and to make translation research in personalized medicine, because what we are talking today are just the general concepts. If you want to do translation research in a daily basis, you have to create infrastructures to do that. I'm not talking about connecting and support infrastructure. You have to do facilities specially devoted to do this. I'm talking about PDX models, patient-derived xenographs. You need to build platforms where you can have the patient there in the hospital, and this hospital is connected with your lab, and you receive samples from this patient that is alive and is taking their drugs, take these primary cultures, build the primary cultures on the lab, treat these cultures that belongs to that patient with another drugs, analyzed pathways, sent back the information to the clinics and said, okay, this patient is not responding anymore, but I tested this or that combination. Why don't we try this? As long as you're using drugs that are approved, you can do this. So this is real translation research, is that you have the patient there, we have the lab here, and then you send the lab back new information to be tested on the patient. This is real translation research. So you have to learn about how to build PDX models, how to build an infrastructure that could be connected with the hospital. It's very risky to try to implement translation research without an hospital nearby or samples nearby. So there's a lot of issues that should be learned if you want to proceed with a career on translation research and personalized medicine, but I can ensure that is an area, a fascinating area for the future. And I hope we can assist to a growing occurring of this kind of formation, specific formation.
Thank you. Any questions? Yes.
Hello. A question. I'm just want to share what opportunities and challenges in the translational medicine. The opportunity I've seen is that it gives us a new way to dig deeper to pathophysiology, and we could know more about diseases and reveal the mystery of human body, and hopefully we could find some new targets to treat, to cure diseases. But the challenge I've seen is that how we could interpret the results. For example, there's a well-known US actress, Angelina Jolie. She took a gene test about breast cancer and decided to have a surgery to prevent cancer. But in my in spite of the gene test, there is no guarantee that breast cancer in the future. I know your opinion on this news.
Okay, thank you. Ver com a situação das mastectomias preventivas.
Prophylacticas. So you are talking about the problem of prophylaxis in breast cancer, right? In hereditary breast cancer. So this is a very serious problem. Of course, that the gene screen that you are talking about will not give all the information that we need. But in case of specifically of breast cancer, when you have mutations in BRCA1 and BRCA2 genes, the penetrance of these mutations for the next generation are quite high. So in these cases, when you do genetic counseling, of course the last word is the patient. She should decide what you're going to do, and their kids, female, will have to do the exam, the screening much earlier than most of the population do, to detect if the mutation is there or not. In some cancers, that is the case of breast cancer, the penetrance of this mutation, which means the consequence of these mutations, are quite high. In these cases, the counseling is to make the prophylactic mastectomy. Today, there are a lot of alternatives, or I would say strategies to minimize the effect of this mastectomy that sometimes been done very early in age. Sometimes girls with 16 or 17 years old has to undergo a mastectomy. But it's really important that people should understand that if they are not doing this, if they decide not to do and take the risk is quite high. It's quite high. For the rest of the gene screens that they are available, like Oncotype DX for breast cancer or Mama Print, we were discussing this today, I don't remember with who, yeah. All these tool-based gene screens are used to give you some help in decision. So just basically they work as a bottleneck for your decision. So they help you to decide. Of course, they won't tell you that is black or white. They will tell you that is gray or darker gray or light gray. And this makes always, every case, to the doctor to tell you what will be the best strategy and account for the risk. That's why there are doctors that only do genetic counseling. And the last word is it belongs to the patient. Of course, that for this kind of tests like Oncotype DX and Mama Print, they have a lot of data that is there that some of this data is not relevant. But one thing is Oncotype DX and Mama Print that basically are useful to drive your decision for a specific therapeutic. They are not used to decide if you're going to do mastectomy or not. The only case you should do mastectomy is if you have a certain type of breast cancer, hereditary breast cancer with certain mutations in BRCA1. And even though there are other polymorphisms that can make this hereditary breast cancer worse than another, and in the end of the day, the decision is always belongs to the patient.
Thank you. So many applications of these new technologies. Okay, if there are no further questions or comments, we will have to end our roundtable discussion and the symposium for this day. I will pass on word to who will. Okay. In a couple of seconds. And I thank you very much for attending and hope that you had at least a flavor of what translational medicine and research is about, and that you get interested in this area and pursue further information and education in this field. Thank you very much.
So I would like to close as well this day of work, the scientific symposium. I wish that you take the most of it, and then you can have new insight for your own reality at your countries on this topic as well. And now I would like to thank you to our speakers for the day and for their good presentations, excellent presentations. And I would like to thank you and deliver the certificate and the gift as well. So I would like to thank to Professor Amir Shahzad, and I'm going to deliver to you. And Professor José Luís Oliveira as well. Professor André Albergaria. And Dr. Emma Paulino. And now, before we continue, I'm just going to pass the word to Angela, that she has some announcements to do. Thank you.
Thank you also from my side again to all the speakers. I think they deserve an applause. My few announcements are partly regarding the regional assemblies, which are scheduled for tonight, because there was some confusion about the time. The regional assemblies will take place from 8 till 10 tonight, not as in the schedule from 10 till 2. From 8 till 10, and all official delegates should attend these sessions. It's not a voluntary session, but as official delegates, you were sent here to be present and to contribute, just as a reminder. Then, as you know from the schedule, tomorrow for the General Assembly, we have the elections scheduled, and we have received a lot of nominations forms, which were already displayed in the back at the entrance and also during the coffee breaks. They were also submitted and are available online. For 2 positions, we have not received any first round nominations. This includes the position of the Chairperson of Professional Development and Chairperson of Pharmacy Education, and therefore nominations are reopened until tomorrow noon, because then we can move on with elections in the afternoon. There will also be an email sent out in a few minutes, which you are highly invited to share, and especially as the contact persons, you should spread the word among your members. Also for the Regional Relations Officer, Regional Projects Officer of the European Regional Offices, we're still waiting for nominations. And for the Eastern Mediterranean Regional Offices, Regional Relations Officer, Regional Projects Officer, and Internal Coordination and Communications Officer. If you have any questions, the call is available online. You can always come and ask if you don't know where to find the information. We are here to help you with everything. Then tonight is the free night, but we also have a special guest here tonight. The ambassador of India will be present here to give a speech in regards to the Congress of the next year. And please stay here in the room until the end of this ceremony. And you're also invited to join the Indian delegation, but I'm pretty sure they will have a word for you and also recall the information and the invitation. Thank you. And now Fahmi will continue with his further announcements regarding the poster competition. Thank you.
Thank you, Angela. So as you may know already, in the back room, we have a scientific poster exhibition. This year we have so many interesting posters and so many great presenters, and we really had a hard time to pick one winner for the competition, but unfortunately we only have one trophy. So I will announce the winner for the scientific poster competition. With the poster title, bupivacaine inhibits endothelin-1 evoked increases in intracellular calcium in model sensory neurons, from Sweden, Malcolm J. Magdesey. I would also invite Professor Amiza Saza to present the trophy for the winner.
Okay. Thank you, everybody.
I also have the trophy for the IPSF critical essay, critical essay competition. And this year, the theme for the essay competition was incorporating translational research training into pharmacy education. But unfortunately, the winner of the competition is not here to attend the Congress. So I will keep the trophy here and send it to him by post. And so, yeah, we will continue the program with the Ambassador of India now.
Good afternoon, everyone. Dear Congress participants, dear ladies and gentlemen, His Excellency and his wife, let me just say a few words. I would like to officially welcome His Excellency Dr. Jitendra Nath Misra and his wife at the 60th IPSF World Congress 2014. It is an honor to have him here with us. As far as my memory goes, this is the first time that an ambassador is attending the World Congress of IPSF. His Excellency is an Indian Foreign Service officer with 32 years of experience in a wide range of diplomatic functions in the United States, the United Kingdom, Israel, Egypt, Bangladesh, Vietnam, Laos, Libya, and Malta, as well as in the Ministries of External Affairs and Defense in New Delhi. He served as First Secretary in Libya and Bangladesh, Deputy Secretary in the Indian Ministry of External Affairs, Joint Secretary in the Indian Ministries of External Affairs and Defense, Additional Secretary in the Ministry of External Affairs, Acting Consul General in the Consulate General of India in Houston, Political Counselor in the Embassy of India in Tel Aviv, and the High Commission of India in London. Also as Consul General in the Consulate General of India in Ho Chi Minh City. He was India's ambassador to the Lao People's Democratic Republic from 2010 to 2014. He assumed charge as India's ambassador to Portugal this year in January 2014. Dr. Misra is an honorary citizen and goodwill ambassador of the city of Houston in Texas. He was awarded the Medal of Ho Chi Minh City and declared a distinguished citizen of the city. For Mauritius Public Service, Dr. Mishra has two commendations from the Government of India. I would now like to welcome him on the stage to address the Congress participants and ask you for another big round of applause.
Namaste. Boa tarde, como está? आप कैसे हैं? انتوا كويس؟ من العرب درست اللغة العربية بالقاهرة منذ زمان. Is there anybody from Vietnam here? No. I'm very impressed by this wonderful gathering. Better than I expected. Mr. Luis Azevedo, Mr. Radoslav Mitura, Dr. Rao V.S.V. Vadlamudi, Ms. Neha Dembla, and friends. I thank you for the invitation to address the 60th World Congress of the International Pharmaceutical Students Federation. I must confess that I have traveled to Porto because of the urging of the Indian Pharmaceutical Association. Since your 61st World Congress will be held in Hyderabad, India, in the year 2015, I am here tonight to support our Indian friends who have come all the way from India to Porto to study the proceedings and promote their own event next year. I wish you the best of luck for the 60th Congress as well as the upcoming one in India. Well, why India? Is it because it's a country of 1.2 billion people? Is it because of the numbers? Or is it something else? Well, talking of the pharmaceutical market, the Indian pharmaceutical market is the third largest in the world in volume and the 14th largest in value. And India will likely be a top 10 market next year. One of the 2 vaccines administered under the United Nations immunization program worldwide is Indian. The world's first dental surgery, in case you don't know, was performed in India. Just as the West has Asclepius, we too have Dhanavantri, the god of medicine. So medical science in India is a very well-developed science historically for thousands of years, as also other natural sciences. Today, I was asked to give an inspirational speech addressing students, a rather unusual experience for me, because most of the time we are addressing adults and policymakers and journalists and professionals. So how do we do this? This is a challenge as well as an opportunity. Being young, it is natural that you are experimenting with life's experiences, always trying out new things, always exploring new terrains, and darting from one thing to another. 650 young people from, I believe, 45 countries, a fascinating mix of the human race from all cultures and creeds. You have a worldwide imprint, doubtless. Pharmacy education is vital in improving human life, and you make a huge difference by bringing your unique knowledge and skills to healthcare. What should I tell you? How do I make an inspirational speech? I don't want to speak about your exams. You'll all do very well. Life is not about technique. It has a much deeper meaning. You did not want a lecture about how to succeed, surely, but something that makes you feel like soaring into the sky, something that makes you take off into the unknown frontiers of knowledge and life, something that inspires you. Can I do it? I don't know. But I guess the best way is to tell you about my own experiences and the way I see life, and do it as honestly as I could. First, being in healthcare, you have a tremendous responsibility to nurture the precious human life embodied in all of us. You are at the confluence of your youth and the coming future, of the young adult and the full-blown one, of memory and expectation, of innovation and experience. You are also confident, mature beyond your years, knowledgeable about the next frontier. At your age, I did not have the good fortune of such optimism. India was a hard country to find work in during the 1970s. And I failed and went on failing. I do not like to talk about failure, except for one reason. Please remember, never forget, that you should never fear failure, because without failure, there can be no success. We humans carry the memory of failure like codes that cannot be broken, but we must confront failure squarely. We cannot be unmade, but we can surely at least correct our mistakes, our failures. That is how you will soar into the sky. When I finished my O-levels at Mayo College in the city of Ajmer, India, I felt a thrill and nervousness at the same time. A thrill because I was entering the next frontier, and nervousness because I was going into the zone of the unknown. How does one organize one's university admission? I did all the work on my own, sent applications to the leading liberal arts schools in Delhi, and virtually camped there in the summer with a relative. It was a great relief to gain acceptance into St. Stephen's College to study history. I prepared early, and I ask you to avoid procrastination. Getting fast off the block is key to success in life. Time is precious, especially your time. As you enter your independent worlds, the protection of home, parents, and siblings will be gone. The loving support of siblings when you are down will be missed. I dealt with this by thinking of my parents. The love that binds family in India is very deep. We dig into our reserves and seek that uncommon solace that sustains us materially and spiritually. Mostly, we succeed. But actually, from my own experiences in the West and East, in the North and South, I believe it is the same everywhere. In the end, it all boils down to education. An educated family makes a huge difference. If you are given the right values, it matters little where you come from. You will cope with this transition well because the love and support of your family remains strong and unconditional. I'm sure of that because all of you come from educated and accomplished families. Never forget how blessed you are to have been born to your loving parents. And that brings me to the big question, the question of identity and community. Who am I? Who are you? Where am I from? Thomas Friedman wrote a book called The World is Flat. Is it actually? He may not have been quite right, but we are bound by shared experiences, by technology, by the internet, by the globalization of music. Think of the Beatles. I love their music so much, and much of the human race likes their music, which touches a deep core in us. Actually, in some ways, I prefer the world to remain round so that we can just go on unraveling its mysteries. Technology and communication may have made our world more flat. Technology is a force for the good, but it can also have negative effects. But why am I speaking about technology to a group that must employ the best technology for better medical solutions? The reason is that we are all susceptible to the lure of technology, especially the young. Modern technology is driven by an undermining automation, with the focus on technique at the cost of imagination, with gadgets, games, sounds, and sights that impair inventiveness. We are persuaded by the absolutist belief in technology's capacity to solve all problems. Our children need cell phones with endless features. We have smart cards to our hotel rooms delivered by neatly dressed front desk staff. The thrill of opening a door with an old-fashioned key is gone. We are trapped in our neediness. Imagine the world in the year 2050. The electronic age would have ushered in the ultimate spell and phrase checks in computers. And then what shall the human mind do? If the computer can do the job, the mind will roam in idleness, and all its creative bursts will be subject to the computer's censorship. We will live in the virtual world of glitz and gadgets. The thrill of writing or receiving a love note with a fountain pen will have gone, and love will become a matter of technique rather than a deeper yearning. So, please do not be seduced by technology, because technology cannot solve all problems. Remember that we also need a clear moral compass to see us not only through our crises and disorders, but also our innermost yearning for love and compassion. Our inner lives remain largely unrealized when we delink them from imagination. You will play huge roles in saving human lives. That is something to accomplish with love and empathy, and not just with medication. I would also like to speak a little bit about culture and identity. When I lived in London, I used to scrounge my pounds to watch theatre at the National. I still remember the two great plays I saw: A Streetcar Named Desire with Glenn Close in it and Iphigenia at Aulis. For sure, that is not all that I did. I also ate fish and chips in ordinary pubs. We need to understand the commonplace to comprehend the more refined things in life. For example, to appreciate a great novel, we need to read a trashy one. Do keep this in mind as you step into the larger world. In many modern industrial societies, we see that the individual is supreme. There is nothing wrong with that. But fellowship and community are also important. The best we humans can do for our fellow humans is to lend a helping hand when we can. In Indian civilization, there is always a place and a second chance for the defeated. I would urge you to remember that in our world, there are many less fortunate people than us. Remember that on our shrinking planet, we are all in it together, and any event anywhere has an impact far away. So many communities and cultures are going extinct in this world of ours, but you are global citizens. Please consider this carefully. The coexistence of cultures and communities is actually vitally important for the survival of the human race. I can only talk of the direct experience of my own country. In India, we have worked out a way to assimilate peoples and cultures for millennia. You may be wondering why I am talking about culture and not medicine. The reason is very simple. The way you live will have a direct bearing upon the way you organize your work. Popular culture is good. By all means, go for it, but never forget the classical cultures of the countries where you locate, and your own countries, of course. In today's age, the move away from classical culture means that everything gets standardized. Please do bear this in mind. There are many things in life beyond MTV. Our belief in a globalized MTV culture will kill art as we know it. There is yet another thing. When I joined the Foreign Service, I got so deeply immersed in work that I forgot about my other identity and existence. Only later was I able to understand that I needed to have a life beyond work. You need to have other interests and a group of friends with whom you don't do business. Even while you are working, you need to do things aside from the business at hand. There is nothing you cannot do, of course. Look at Bill Gates. He had a humble origin, but he rose to global eminence. Look at the great Indian entrepreneur Mohan Singh Oberoi. He was once a bellboy at the Clark's Hotel at Shimla, India. He rose to head one of the biggest hotel chains in India. I myself was born in a village, but I got a scholarship to study in the most expensive and the best boarding school in India. I was not privileged. You are privileged. The question is what you do with being privileged. Remember that your origins matter little. Most of you will work in developed industrial meritocracies. I taught at Georgetown University, Washington, and I can tell you that who you are matters little over there. Please do remember this. Once you go into the larger world, you are on your own, and you will be judged by how well you perform. The more you study, the less you know. Ask the Vietnamese I used to meet in the park during my tour of duty in Ho Chi Minh City, who, even though I did not know them, went on speaking to me in English to improve their language skills. It is all a question of attitude. In this context, I want to say a bit about aspiration and expectation. Aspirations are good, but expectations are something different. An aspiration can be measured against one's own set of goals and is driven by one's own impulses, but expectation could also involve someone else. Please do keep this in mind. Both aspiration and expectation begin with baby steps. Unless we dare to dream, we can never get what we want. Ask Professor Abdul Kalam, the former President of India, who has famously written about igniting the Indian mind so that India can catapult itself into the 21st century as a developed country in 20 years. Let me now turn to mundane matters. I suggest that you learn some cooking. Why am I talking about cooking? Because mom won't be around forever to do it for you. You are mostly 22, 23, 24. When I was alone with my older son at Washington, I cooked because eating out all the time was not an option. My son also learned to cook, and this saw him through university. I know that for a young adult, cooking is hard, but it is well worth the effort. Please avoid junk food as far as possible because the after effects will haunt you in later life. Being in the medical profession, you know this very well, of course. So, in conclusion, you are ready to take on the world. I'm sure you will excel in whatever you do. With the superb education you have had, in whichever continent you come from, you can accomplish anything. You are on the verge of making careers in a profession where you will not only nurture human lives, but save them. What could be more privileged? I don't think anything than saving precious lives. There are not many better career options, therefore. So, steel yourselves for the challenges that await you, but always enjoy what you do. So I congratulate you on your 60th Congress once again. Thank you. Yes, yes.
Thank you so much, Ambassador Sir. This was certainly a very, very inspiring and motivating speech. I'm sure you all enjoyed it, and it was certainly very touching for us, for him to come and address you all on his behalf and to invite you and help you enjoy here. Now, I would like to request our Chief Advisor, Mr. Murthy Chatturi, and Madam Excellency on stage. We would like to have a very small pinning ceremony. Let's have a round of applause. I would also like to invite the chairperson of IPSF, Mr. Radoslav Michura, and ma'am. Let's have a round of applause. I would request Mr. Radoslav Michura to pin the ambassador with the IPSF pin. This is certainly a moment of joy and pride for all of us. I would now request my delegation from India to be here on stage and make this memory in pictures forever. Our Vice Chairperson, Mr. Vikas, will present a small gift to the ambassador before this happens. This is our idol, Ganpati. Thank you, Ambassador, sir. Thank you, madam. I would now request His Excellency, Ambassador, sir, and Madam Excellency to proceed for a small reception that we have arranged for them. The students are requested to wait till they proceed, and then we can have our further announcements. Are you all ready for the Indian night? So guys, for those of you who don't know, we have an Indian night at the Faculty of Pharmacy arranged in the gardens. I would now request, we would have a dance party there and we would have, of course, everything arranged there for you. I hope you all are ready for it. I can see people excited already. It's a dance party and we would have a dance of Dandiya and Garba. Just for you to know, it's one of the traditional dances of India. And it is based on, it is a very multicultural dance as it is played in various parts of India. It originated from a war between a goddess and a demon. And now it is symbolic of all the fight that happened between them. I would request you to not have those fights and not hit each other during that. We would certainly help you. We will first teach you the dance and we would expect you to enjoy after you all know the dance. The arrangements are made there and you all need to be really a little disciplined, let me say. I know it's a party, but at least when you're going there, we would have the arrangements and we would tell you right there how and when to play the dance. Is it okay? India is ready. Are you? Louder. Thank you.
So guys, before we leave, I just have an announcement about the bus. So there will be a bus at 8:15, okay, to take the participants of residence number 3 to their residence. But it's only one, okay, because it's free evening, so you don't have any more buses, okay? So just try to manage your time. So yeah, and enjoy yourselves this evening, and bye. See you tomorrow at 9.